You've probably seen bits and pieces of this story before. A documentary about dinosaurs here, something about early humans there, maybe a segment on the Cambrian explosion if you've gone down a YouTube rabbit hole at 2 a.m. But here's the thing nobody tells you. These pieces aren't random. They're all connected. They're chapters in the same book, and that book is about you. See, most of us learn evolution like we're looking at a photo album someone dropped down the stairs. We pick up a picture here or a picture there and try to figure out what order they go in. And sure, each photo is interesting on its own, but we never see the whole album arranged the way it's supposed to be. That's what this is, the whole album, in order from the first page to the last. And here's what makes it different from everything else you've watched. Every single organism we're going to talk about is your direct ancestor, not a cousin species, not an interesting side branch, not something kind of like your ancestor, your actual literal family line. The chain that connects you to the very beginning of life. Think about that for a second. 4 billion years of parents having children, an unbroken line stretching back to the first things that could make copies of themselves. If that chain had broken, even once, if just one of your ancestors had died before having offspring, you wouldn't be here, none of us would. We've spent months on this, not because evolution is complicated, though it is, not because we wanted to make the longest documentary possible, but we might have. We spent months to put the complete story in one place like no one has before. And there's a reason for that. The scientists who study the origin of life speak a completely different language than the scientists who study dinosaurs. The people who study dinosaurs barely talk to the people who study early humans. Everyone knows their specialty incredibly well, but almost nobody has traced the entire path from chemistry to you without skipping anything. We're going to do that, together. And, by the end, you won't just have a vague sense that evolution happened. You'll have seen exactly how it happened, step by step, with no gaps, no jumps, and no hand-waving. This is your story, your deep history, the longest and most unlikely success story ever told. Let's see where you came from. Earth's surface holds the same basic ingredients found everywhere in the universe. Hydrogen, carbon, oxygen, nitrogen, phosphorus, sulfur, and scattered metals like iron and nickel. These atoms don't stay separated for long. In early oceans, in volcanic gases, and on wet rock surfaces, they constantly bump into each other and stick together in simple combinations. Volcanoes release carbon dioxide, water vapor, nitrogen gas, hydrogen sulfide, and traces of other gases into the air and water. Lightning strikes these gas mixtures thousands of times daily. Each bolt breaks molecules apart, like snapping apart linked chains, leaving fragments that immediately search for new partners. When carbon dioxide breaks, it releases carbon and oxygen pieces. When nitrogen gas splits, the nitrogen atoms grab hydrogen to form ammonia. Water molecules break into hydrogen and oxygen fragments. These pieces recombine in the cooling air and in rainwater, forming small but important molecules like hydrogen cyanide, carbon monoxide, and formaldehyde. Think of lightning as a factory that takes stable, boring molecules and shakes them into reactive pieces that can build something more interesting. The products wash into pools, soak into mineral surfaces, and dissolve into the ocean. At the ocean floor, a completely different process runs continuously. Seawater seeps down through cracks in the crust, heats up, and rises back through metal-rich rock. The hot water dissolves iron and nickel that shoots back up as underwater hot springs. When this metal-rich fluid meets cold seawater, the dissolved metals precipitate out as fine particles, building chimney-like vents. These metal surfaces act like workspace counters in a kitchen. They hold ingredients in place long enough for them to connect. For example, carbon dioxide from seawater lands on hot iron sulfide surfaces, picks up hydrogen atoms from the water, and transforms into formate and formaldehyde. Hydrogen gas produced where hot rock meets seawater splits on the same surfaces, leaving hydrogen atoms loosely attached. When carbon monoxide lands nearby, it grabs these hydrogens one by one, building longer and longer carbon chains. Some of these chains pick up an oxygen at one end, creating fatty acids, molecules 8 to 16 carbons long with one end that loves water and one tail that avoids it. When enough fatty acids accumulate in water, they spontaneously arrange themselves into bubbles. It's like a drop of oil in water, naturally forming spheres. The water-loving ends face outward, touching the water, while the water-hating tails cluster together away from it. These bubbles, called vesicles, form closed spheres with water trapped inside. They're the first hint of inside and outside on a planet where everything seems to be mixed together. Meanwhile, in shallow pools where water collects and evaporates, hydrogen cyanide from lightning-struck air accumulates. When 5 hydrogen cyanide molecules link together in the right sequence, they form adenine, one of the bases that will later appear in RNA. It's a bit like 5 identical Lego bricks that can only snap together one way to make a stable structure. The process works best when molecules are concentrated by evaporation, forcing them into close contact. Amino acids, the building blocks of proteins, form through several routes simultaneously. The simplest pathway starts with an aldehyde, any small molecule with a carbon double bonded to oxygen and a hydrogen. When this aldehyde meets ammonia in water, they connect, temporarily linking nitrogen to the carbon. If hydrogen cyanide then adds to this combination, it attaches a cyanide group. Finally, when water reacts with this cyanide group over time, faster when warm, it converts into a carboxylic acid group, creating an amino acid. The sequence produces glycine from formaldehyde, alanine from slightly larger aldehydes, and serine from aldehydes that have an extra oxygen. The process works in any pool where aldehydes, ammonia, and hydrogen cyanide exist together, which means it works almost everywhere, in pools on mineral surfaces and near vents. It's like having a recipe that works in any kitchen as long as you have the 3 basic ingredients. Space contributes too. When meteorites are broken open in laboratories, they contain dozens of amino acids, including all the common ones and many strange ones never used by life. These amino acids formed in asteroids and comets through similar chemistry, then arrived on Earth with every impact. Over millions of years, the delivered amount adds up substantially. Most amino acids exist in 2 mirror image formats, like left and right hands. Simple chemistry makes equal amounts of both, but small pushes can break this balance. Some types of light spiral as they travel, like a corkscrew, and this spiraling light destroys one mirror form faster than the other. Certain mineral crystals' faces hold one form more tightly than the other. During repeated cycles where water evaporates and returns, the form that sticks to the mineral surface stays protected while the other breaks down in solution. After thousands of cycles, one form dominates. It's like a biased coin that lands heads 51% of the time. After a million flips, heads clearly wins. Sugars form when formaldehyde molecules, already present from atmospheric and vent chemistry, link together. 2 formaldehydes make a 2-carbon sugar. That sugar plus another formaldehyde makes a 3-carbon sugar. The process continues, building 4 and 5-carbon sugars, including ribose, the sugar backbone of RNA. But sugars are fragile. The same features that let them form also make them break apart quickly in water, sometimes within hours in warm conditions. 2 factors rescue them. Borate minerals, found in volcanic deposits and evaporate beds, grab onto ribose and hold it in a protective shape that extends its survival from hours to weeks. It's like wrapping a delicate glass ornament in bubble wrap. Freezing also helps. When a pool freezes, dissolved molecules concentrate in thin liquid pockets between ice crystals. The cold slows breakdown reactions, while the high concentration speeds up formation, and molecules that form in these pockets last longer than they would in warm, dilute water. Glyceraldehyde, one of the 3-carbon sugars formed in this process, can transform into glycerol, where it encounters metal surfaces that supply hydrogen. The result is a more stable 3-carbon molecule with a hydroxyl group on all 3 carbons. When glycerol and fatty acids meet in a drying pool, they can link together, forming molecules that make even more stable vesicles than fatty acids alone. The hardest step is assembling complete nucleotides, the building blocks of RNA. Each needs a base, a sugar, and a phosphate group joined in a specific arrangement. The difficulty isn't making the pieces separately, but connecting them without everything falling apart immediately. Phosphate usually sits locked in insoluble minerals. However, meteorites bring a reactive phosphorus mineral called schreibersite that dissolves and releases phosphate in forms that stay available longer. In drying pools on volcanic surfaces, small molecules concentrate as water evaporates. Glycolaldehyde, glyceraldehyde, cyanamide, and phosphate all squeeze together in a thin film. When ultraviolet light hits this film, unfiltered by any ozone layer, it drives reactions that wouldn't happen in ordinary water. Rather than building free ribose and then trying to attach pieces to it, these molecules react together in a coordinated sequence. Glycolaldehyde and cyanamide form an intermediate structure. This reacts with glyceraldehyde that has already phosphate attached. Through several rearrangements driven by UV light, the result is nucleotide-like molecules where the phosphate is in a reactive form, a coiled spring ready to link with the next nucleotide. The key is that the sugar, base fragments, and phosphate build up together instead of trying to force 3 separate finished pieces to connect. Clay minerals like montmorillite help these nucleotides link into chains. The clay's flat surfaces carry a slight charge that attracts nucleotides. The surface holds them in organized rows, and when water evaporates, the nucleotides connect into short RNA-like chains. Laboratory experiments show chains up to 50 units long can form this way through repeated additions and drying cycles. 3 physical patterns repeat everywhere. Concentration brings molecules close enough to react, achieved by evaporation, freezing, or sticking to surfaces. Activation overcomes barriers that prevent reactions in pure water, provided by reactive chemical groups, mineral catalysts, or UV light. Cycles drive progress through repetition, wet, dry, hot, cold, light, dark sequences that favor formulation during one phase and prevent breakdown during another, letting products accumulate over many repetitions. After hundreds of millions of years, Earth's surface holds a rich chemical inventory spread across countless pools, shorelines, vents, and mineral surfaces. Amino acids exist everywhere, with some locations showing more left-hand forms. Sugars include ribose, appear wherever formaldehyde chemistry operates, and borate or cold protects them. Fatty acids form vesicles anywhere their concentration crosses the self-assembly threshold. Glycerol mixes with fatty acids in the same environments. The 4 nucleobases, adenine, guanine, cytosine, and uracil, coat mineral surfaces and concentrate in pools. Activated nucleotides form in smaller quantities wherever the right precursors, minerals, and light combine. Short chains of amino acids and nucleotides form on surfaces and in drying films, accumulate, break apart, and reform. Vesicles drift through water, trap whatever molecules happen to be nearby when they form, and sometimes split when stretched. Clay surfaces hold organized arrays of small chains. Nothing copies itself yet. Nothing maintains its composition against the surroundings. But the planet now holds, scattered across many locations, all the molecular types that life will later use, along with natural processes that concentrate them, link them into chains, and enclose them in compartments. The stage is set for these pieces to start interacting in new ways, for chains to work with surfaces, for chains to interact with compartments, for chains to affect other chains. What comes next is no longer just making building blocks, but watching those blocks stumble into the first hints of organization. The building blocks now exist everywhere. Amino acids, nucleotides, fatty acids, and glycerol scattered across pools, shorelines, and ocean floors. But life doesn't run on isolated building blocks. It runs on chains. Proteins made from hundreds of amino acids, RNA made from thousands of nucleotides, membranes made from organized sheets of lipids. The question isn't whether these pieces exist. The question is how random molecules floating in water become organized structures. The answer is brutally simple. They can't, just not in water. Amino acids have 2 ends, an amino group on one side and a carboxyl group on the other. Bring them together and they can link, releasing water and forming a peptide bond. Except in water, the reaction runs backwards just as fast as forward. Water molecules constantly attack any bond that forms. You're trying to build chains in the exact solvent that destroys them. Unless you get rid of the water. Picture a tidal pool at dawn. Amino acids drift freely, 1 per million water molecules. The sun rises. Water evaporates. By noon, concentration has jumped a millionfold. There's barely any water left, just a thin film of molecules pressed together. The chemistry flips. Without bulk water attacking bonds, amino acids link spontaneously. 2 units, 3, 5, 10. The tide returns at sunset, but the chains persist because breaking them requires water molecules hitting at precisely the right angle. That happens, but slowly. The next day, the cycle repeats. After 100 cycles, some peptides reach 30 amino acids long. Scientists have replicated this in labs. Wet-dry cycling on mineral surfaces produces peptides spontaneously. Early Earth had billions of tide pools cycling daily for hundreds of millions of years. These early peptides aren't proteins, but they're different enough to matter. Some stick to mineral surfaces, concentrating where they'll participate in the next reaction. Some migrate to oil-water boundaries where membranes were formed. And some, purely by accident, grip metal ions like iron at specific angles. That changes local chemistry. Water molecules bound to that iron become acidic, eager to break specific bonds. Reactions that would take years happen in days. It's crude, maybe 10 times faster instead of a millionfold, but it crosses a threshold. A molecule that changes what happens around it, that's the seed of catalysts. RNA chains face the same problem but solve it differently. The key is clay. Montmorillonite clay has negatively charged surfaces. Activated nucleotides carry positive charges. Opposite charges attract, so nucleotides stick to clay in organized rows. When water evaporates, nucleotides press together. Phosphate groups link to sugars. One dry cycle will give 2 unit chains, next cycle would be 3, and after a dozen cycles, chains could reach up to 50 nucleotides long. Scientists have watched this happen, activated nucleotides on Montmorillonite wet-dry cycling with no enzymes required. But length isn't what makes RNA interesting, sequence is. String together random nucleotides and you get a floppy chain. But certain sequences fold. When one part bends back and meets another, complementary bases, A with U and G with C, form hydrogen bonds. The chain locks into a specific three-dimensional structure and creates a shape. And the shapes can do things. Some RNA sequences fold into structures, creating pockets, like tiny chambers enclosed by the backbone itself. A molecule drifting into the pocket experiences chemistry nothing like bulk in water. Maybe the pocket excludes water. Maybe it holds magnesium ions at a precise angle. Maybe it traps 2 molecules that would never meet otherwise. But by pure accident, some folded RNA makes specific reactions happen 100 times faster. Over geologic time, across trillions of pools, that's revolutionary. Here's the critical discovery. Some RNA shapes can link nucleotides together. An RNA chain folds into a pocket with precise geometry. Binding sites, positioning mechanisms, chemical groups help phosphate bonds form. This RNA is building more RNA. It's slow, error-prone, and mistakes happen every 10 to 20 nucleotides, but it works. Even better, RNA can template its own copying. Bases sit exposed like a pattern. Free nucleotides drift past. So when a complementary base approaches, hydrogen bonds hold it temporarily. If a catalytic pocket assists, a new base adds to the growing copy. One nucleotide at a time, a complementary strand assembles. The copy isn't perfect, but imperfect copying beats no copying. Sequences folding into a stable shape survive longer, become more common, and sequences with catalytic pockets copy themselves faster and dominate the population. Random sequences break down and vanish. This isn't full evolution, there's no inheritance system yet, but it's the first filter where structures determine survival. And we know RNA can do this because RNA molecules with these abilities exist right now, ribozymes, your ribosome. Scientists have created RNA copying sequences 95 nucleotides long. The chemistry works. Meanwhile, peptides and RNA discover each other. RNA carries negative charges. Peptides with positive amino acids get pulled towards the RNA electrostatically. Some peptides wrap around RNA like protective sheaths, extending its lifetime from days to weeks. Some slip into RNA-folded pockets, adding chemical groups RNA lacks. The combination outperforms either molecule separately. RNA provides structural precision, and peptides provide chemical versatility. This partnership isn't modern, it's ancient. Your cells still run on it. fatty acid vesicles start trapping these systems inside. Most vesicles trap nothing useful, but near clay beds in drying pools, some close around functional molecules, and confinement changes everything. In the open ocean, a catalyst product disperses immediately. Inside a 1 micrometer vesicle, molecules encounter each other thousands of times. A catalyst works on the same substrates repeatedly. Products build to useful concentrations. If the catalyst produces something helping the vesicle, that benefit stays with the structure that made it. The membrane isn't sealed. Small molecules can still slip through, but large chains can't escape. Raw materials enter, but complex products accumulate. The membrane grows as fatty acids insert themselves. Eventually, the vesicle destabilizes. It's just simply too large. Physical forces stretch it into a dumbbell, the neck pinches, and 2 smaller vesicles form. Each daughter inherits half the parent's contents. If the parent held catalytic RNA, protective peptides, and raw materials, those divide as well. The RNA keeps functioning in both compartments. Over hundreds of generations, vesicles with functional contents become common. Vesicles without disappear, which is automatic filtering. One final ingredient is metabolic cycles. Near hydrothermal vents, thioesters form, molecules storing chemical energy. They're unstable, eager to transform carbonyl groups. Hit an amino acid with a thioester, and you get an activated amino acid that can join chains without external energy. Chain these reactions together, and you get a cycle. Simple molecules entering through the membrane transform into complex molecules trapped inside. Some cycles are autocatalytic. They produce something accelerating their own operation. Chemistry that sustains itself and improves. By now, Earth holds pockets where chemistry has become organized. Tidal pools where peptides grow daily, clay beds where RNA copies itself, surface waters where vesicles divide, vents where cycles turn simple inputs into complex outputs. None of these are alive. They don't maintain themselves indefinitely, and don't copy reliably, don't adapt purposefully, but they aren't random. What brings them together is location and time. A vesicle forms in a pool containing both peptides and RNA. Both get trapped, the peptides protect the RNA catalyzes reactions helping peptides grow. Together, they produce lipids helping membranes expand. This combination persists longer and divides more often. After thousands of generations, descendants resemble parents more than chance predicts. Combinations that cooperate spread, combinations that interfere vanish. Earth now holds compartmentalized systems where useful molecules find each other repeatedly, where functional products accumulate, and where successful combinations pass something resembling design to descendants. Not life yet, but standing at the threshold. One step remains: converting the loose correlation between RNA sequence and catalytic function into a systemic code. That step transforms organized chemistry into biology. Vesicles now drift through warm surface waters, some empty, some carrying RNA or peptides trapped inside. Most do nothing, just passive bubbles that form, float, and eventually break apart. But a few contain something different, RNA that can catalyze reactions. These rare vesicles become the first chemical systems that might eventually be called alive. The transformation happens when a vesicle encloses RNA capable of building more RNA. Activated nucleotides, small enough to slip through the membrane, drift inside. The catalytic RNA grabs them and links them together. New chains accumulate because they're too large to escape. If one new chain folds into a catalytic shape, now there are 2 RNA molecules building more RNA. The vesicle becomes a factory. This creates the first meaningful difference between vesicles. Empty ones drift passively. RNA-filled ones grow because accumulated chains push outwards on the membrane. The membrane responds through simple physics. Stretched lipid bilayers have gaps where new fatty acids insert from surrounding water, expanding surface area. Eventually, the vesicle becomes mechanically unstable, too large, wrong geometry. A current deforms it into a dumbbell. The neck pinches, and 2 smaller vesicles form, each inheriting roughly half the parent's RNA. Jack Skozak's lab at Harvard demonstrated this exact process. They created protocells containing RNA, provided activated nucleotides, and watched the vesicles grow and divide mechanically. No genes controlling it, no proteins coordinating it, just physics and chemistry. Over hundreds of generations, RNA-containing vesicles outnumber empty ones because the former divide more often. Automatic filtering, the ones that work persist, and the ones that don't disappear. But there's a problem that nearly ends everything. Copying isn't accurate. Primitive ribozymes make roughly one error every 50 to 100 nucleotides. After 10 rounds of copying, most descendants are garbage. Wrong bases everywhere, broken chains, non-functional shapes. A ribozyme producing 9 useless copies and 1 functional one should be doomed. Errors accumulate faster than selection can fix them. This is called error catastrophe, and should make molecular evolution impossible. But here's why it doesn't destroy the system. Competition within finite space. A vesicle 1 micrometer across holds roughly 1 femtoliter of water. If that vesicle contains 1,000 RNA molecules competing for perhaps 10,000 activated nucleotides entering per hour, copying speed matters. An RNA sequence copying itself in 2 hours claims more resources than one taking 4 hours. After 10 generations, the faster copier produces 10,024 descendants, while the slower produces only 59. The faster sequence now controls 95% of the vesicle's RNA. But speed isn't enough. An RNA copying fast but making errors every 20 nucleotides produces mostly garbage. An RNA copying slowly but accurately, only 1 error per 200 nucleotides, maintains functional sequence better. The optimum isn't maximum speed, it's the right balance between speed and accuracy. Gerald Joyce's lab at Scripps evolved RNA molecules that improved their copying fidelity from 1 error per 50 bases to 1 error per 200 bases through selective pressures alone, proving selection can favor accuracy when accuracy demonstrates survival. Some RNA sequences discover something even more valuable. They catalyze reactions helping the vesicle itself. In 2013, Sheriff Manzi's lab in Italy showed certain ribozymes can catalyze fatty acid synthesis inside vesicles. When RNA produces fatty acids that insert into the membrane from inside, the membrane grows faster. That vesicle divides every 12 hours instead of 24, leaves twice as many descendants, and the RNA sequence spreads. The RNA that helps its container wins. Now the crucial transition begins. RNA starts directing peptide assembly. Some RNA sequences grip amino acids in specific pockets through chemical complementarity. A pocket shaped to fit lysine long, positively charged side chain holds lysine better than other amino acids. No code yet, just physical fit. If the RNA pocket positions 2 amino acids close together, they link, forming a dipeptide. Different RNA shapes produce different dipeptides. Here's what makes this revolutionary. RNA sequence determines pocket shape, which determines which amino acids fit, which determines what peptides get built, which determines what the peptide does chemically. The transition to true coded translation happens through adapter molecules emerging gradually. Some short RNA chains, 10 to 15 nucleotides long, fold into shapes with 2 distinct ends. One end binds a specific amino acid through chemical affinity. The other has a sequence that base pairs with complementary sequences on other RNAs. These proto-transfer RNAs create a physical link between RNA sequence and amino acid identity. The critical insight is that proto-tRNAs aren't irreducibly complex. Each function existed separately before combining. RNA molecules that bind amino acids already existed. Many ribozymes used amino acids as cofactors. RNA molecules that recognize specific sequences through base pairing already existed. That's just how copying works. But a molecule doing both isn't a leap. It's just combining the 2 existing capabilities. The first proto-tRNAs were probably terrible at both jobs, binding amino acids weakly, recognizing sequences imprecisely, but even 10% specificity beats a random incorporation. Peptides made with this system would have certain amino acids appearing more often at certain positions than chance predicts. Any improvement in specificity, tighter amino acid binding, more accurate sequence recognition, gets selected because better correlation means more reliable production of useful peptides. Earth held perhaps 10 to the 22nd power protocells at any time during this period. Each divided roughly daily. Over 100 million years, that's 10 to the 30th evolutionary experiments. The proto-tRNA innovation, however improbable for a single protocell in a single generation, only needed to happen once. With 10 to the 30th trials, even 1 in a trillion events become statistical certainties. Peptides transform protocell capabilities. Some peptides stabilize RNA, extending functional lifetimes from hours to days. Some catalyze reactions RNA handle poorly. Some interact with membranes controlling what passes through. Each function helps the vesicle, helps the RNA that produced the peptide, and spreads that RNA sequence through descendants. RNA dominates these early systems because it can store information like DNA and catalyze reactions like proteins. But RNA has crippling limitations. It breaks down in water within hours or days, and it's mediocre catalyst compared to proteins. The solution is division of labor, emerging gradually. Some ribozymes evolve the ability to chemically modify nucleotides before incorporating them. One crucial modification removes one oxygen atom from ribose sugar, creating deoxyribose. But why would this mutation be selected for before DNA's advantages are obvious? Well, because even mixed chains benefit immediately. An RNA chain with 10% deoxynucleotides lasts roughly 30% longer before breaking down. If that RNA is catalytic, longer lifetime means more work done, means more products made, means more benefits to the vesicle. The advantage is immediate, even for hybrid chains. Over time, protocells evolve ribozymes, producing deoxynucleotides more consistently. Eventually, chains made entirely from deoxynucleotides, DNA, serve as stable backup storage while RNA continues as working copy. This arrangement solves error accumulation. RNA copying RNA propagates errors forward, with each copy templating the next generation. But if DNA serves as a master template and fresh RNA copies are made from DNA each generation, errors in RNA copies don't compound. Only errors copying DNA matter in long term. DNA copying can be slower and more careful because it happens less frequently. Mutation rates drop from 1 per 100 nucleotides to 1 per 10,000 nucleotides. Information preservation becomes possible. By this point, protocells contain information stored in DNA, working copies in RNA, functional molecules as peptides, membranes that grow and divide, and primitive metabolism producing useful products from raw materials. They maintain themselves against entropy. They reproduce. They pass information to offspring. Success depends on how well their molecular contents cooperate. This is the edge of life. They still lack regulated metabolism, sophisticated membrane control, or complex genetic codes, but they have the core properties: self-maintenance, reproduction, information transfer, and improvement through selection. Everything afterwards is elaboration. The planet now holds systems that don't just exist. They persist, adapt, and evolve. This chapter describes the single most important event in our lineage's history. Every complex organism alive today, every animal, every fungus, every plant, every protist, descends from one extraordinary merger that happened roughly 2 billion years ago. An archaeal cell engulfed an aerobic bacterium, and instead of digesting it, the two established a permanent partnership. The bacterial cell became the mitochondrion, the energy-producing structure found in virtually all eukaryotic cells, including ours. This event, called eukaryogenesis, transformed a simple archaeal cell into something radically different, capable of complexity impossible for any prokaryote. The exact circumstances of this merger remain debated, but the broad outline is clear. An archaeal cell, likely an Asgard archaeon living in sediment or near hydrothermal environments, encountered an aerobic bacterium, probably an alpha-proteobacterium capable of using oxygen to extract energy efficiently. The archaeal cell somehow took the bacterium inside. This might have happened through direct engulfment if the archaeon already had primitive abilities to bend and manipulate its membrane. Or it might have started as a different relationship. Perhaps the bacterium was initially a parasite, invading the archaeal cell, or perhaps the archaeon was feeding on bacterial waste products and the bacterium took up residence nearby, gradually getting closer until it ended up inside. However it began, The crucial point is that the bacterium survived inside the archaeal cell and kept functioning. It continued performing aerobic respiration, using oxygen to break down organic molecules and generating ATP. The ATP it produced leaked out and became available to the host cell. This gave the host archaeon access to aerobic metabolism without having to evolve all the complex machinery itself. The host provided the bacterium with a protected environment and a steady supply of organic molecules to metabolize. Both benefited enough that the partnership persisted. At first, the relationship was probably loose and unstable. The bacterium retained all of its genes and could theoretically survive independently if separated from the host. But over time, through millions of years, the partnership deepened. Some bacterial genes transferred to the archaeal cell's chromosome. This happens occasionally through horizontal gene transfer and provides a backup copy if the bacterium's own chromosome degrades. Once a bacterial gene existed in the host genome, the host could produce that protein and import it into the bacterium. The bacterium no longer needed its own copy of the gene, so mutations could disable it without consequence. This process repeated hundreds of times. One by one, bacterial genes transferred to the host's genome were expressed by the host, and their protein products were imported back into the bacterium. The bacterium's genome shrank as genes became redundant. Modern mitochondria retain only a tiny fraction of their ancestral bacterial genes. Humans have just 37 genes left in mitochondrial DNA, encoding only the most essential proteins that must be produced locally. Everything else the mitochondria needs, roughly 1,500 different proteins, is encoded in the nucleus, produced in the cell's main compartment, and imported into the mitochondrion. This gene transfer locked the partnership permanently. The bacterium could no longer survive independently because it lacked most of the genes needed for a free living existence. The host could no longer survive without the bacterium because it depended on mitochondrial ATP production. They had become a single organism, a eukaryotic cell, with two genomes working in coordination. The energy advantage this provided was transformative. Aerobic respiration extracts roughly 15 times more ATP from the same organic molecule than anaerobic fermentation. A eukaryotic cell with mitochondria had access to vastly more energy per unit of food than any prokaryote relying on fermentation or on the less efficient anaerobic metabolic pathways. This surplus energy enabled things impossible for prokaryotes. The first major consequence was size. Prokaryotic cells are limited to a few micrometers in diameter because they generate energy at their membranes. As a cell grows, its volume increases faster than its surface area. A larger cell has proportionally less membrane surface relative to its interior volume, which limits how much energy it can produce per unit of cellular material. This creates a hard size ceiling. Prokaryotes can't grow much larger than a few micrometers without starving their interiors of energy. Eukaryotic cells bypass this constraint by internalizing energy production. Mitochondria are internal structures, each with their own membranes generating ATP. A eukaryotic cell can pack its interior with mitochondria, scaling energy production with volume instead of being limited by surface area. This allows eukaryotic cells to grow much larger, 10 to 100 times the diameter of typical prokaryotes, without hitting energy constraints. Larger size enabled internal complexity. A bigger cell has more internal volume to organize into specialized regions. Early eukaryotes developed internal membrane systems, folds and compartments separating different functions spatially. One region might specialize in protein synthesis, another in breaking down molecules, another in storing materials. This compartmentalization improved efficiency because specialized regions could maintain different chemical conditions optimal for different processes. The nucleus evolved as part of this compartmentalization. In prokaryotes, DNA floats in the cell interior, and ribosomes start translating RNA into protein while the RNA is still being transcribed from DNA. Everything happens simultaneously in the same space. In eukaryotes, DNA became enclosed in a membrane-bound nucleus. Transcription happens inside the nucleus, producing RNA. The RNA then travels out through pores in the nuclear membrane to reach ribosomes in the cytoplasm, where translation into protein occurs. This separation between transcription and translation opened new possibilities for regulation. Inside the nucleus, RNA can be modified before it leaves. Sections can be cut out and the remaining pieces spliced together in different combinations, producing multiple different proteins from a single gene. Chemical modifications can be added that affect how the RNA behaves. The RNA can be checked for errors and corrected before translation begins. None of this is possible in prokaryotes, where transcription and translation are coupled. The nuclear membrane created time and space for information processing between reading the gene and producing the protein. Another consequence of eukaryogenesis was the evolution of a sophisticated cytoskeleton. Archaea had primitive versions of cytoskeletal proteins, but eukaryotes elaborated these into complex dynamic networks. The eukaryotic cytoskeleton consists of three main types of filaments: microtubules, actin filaments, and intermediate filaments, that crisscross the cell interior, providing structural support, enabling shape changes, and serving as tracks for molecular motors to transport cargo. The cytoskeleton allows eukaryotic cells to do things prokaryotes cannot. They can actively change shape, extending protrusions to explore their environment or engulf particles. They can transport materials long distances within the cell by loading cargo onto motor proteins that walk along cytoskeletal tracks. They can organize their interior, positioning organelles in specific locations and moving them when needed. The cytoskeleton essentially transformed the cell from a static bag of molecules into a dynamic, organized, and responsive system. These capabilities fed back into enhancing the eukaryotic lifestyle. The ability to change shape and engulf particles enabled phagocytosis, actively hunting and consuming other cells. Eukaryotes became predators, eating bacteria and other small organisms wholesale rather than just absorbing dissolved nutrients. This opened entirely new ecological niches and intensified selection for traits that improved hunting, evading predators, or competing with other eukaryotes. The endomembrane system, an interconnected network of membrane compartments including the endoplasmic reticulum, Golgi apparatus, and various vesicles, evolved to manage the traffic of proteins and other molecules within the cell. Proteins destined for different locations or functions could be synthesized, modified, packaged, and delivered precisely where needed. The cell gained logistics, the ability to manufacture, modify, and deploy thousands of different molecular products in an organized way, rather than dumping everything into a common pool. Sex evolved relatively early in eukaryotes, though its origins remain somewhat unclear. Prokaryotes exchange genes horizontally, but they don't have true sexual reproduction where two cells fuse, their genomes combine, and the mixture is then divided up among offspring. Eukaryotes developed meiosis, a specialized cell division that halves the chromosome number, and fertilization, where two cells fuse and their chromosomes combine. This creates offspring with genetic material from two parents, shuffling genes into new combinations each generation. The advantage of sex is genetic variation. Asexual reproduction produces offspring genetically identical to the parent, except for rare mutations. Sexual reproduction combines and recombines genes, producing offspring with different combinations of traits. In a changing or competitive environment, this variation means some offspring will be better suited to current conditions than either parent. Over time, sex allows faster adaptation and more effective responses to environmental challenges and to other evolving organisms, particularly parasites and predators. By about 1.5 billion years ago, eukaryotic cells possessed all these features: mitochondria providing abundant energy, a nucleus separating transcription from translation, an extensive endomembrane system organizing internal logistics, a dynamic cytoskeleton enabling movement and shape changes, the ability to engulf and consume other cells, and sexual reproduction generating variation. These weren't minor tweaks to the prokaryotic design. They represented a fundamental reorganization of cellular architecture, possible only because of the energy surplus mitochondria provided. Our lineage at this point consisted of single-celled eukaryotes, probably living in aquatic environments, feeding on bacteria and possibly on other smaller eukaryotes. They were larger, more complex, and more dynamic than anything that had existed before. But they were still solitary cells, each living independently, reproducing by dividing, with no coordination between cells beyond competition and predation. The transition to multicellularity, cells staying together after division and coordinating their behavior, was the next major step, but that required additional innovations beyond what eukaryogenesis provided. Cells needed ways to stick together reliably. They needed communication systems allowing cells to coordinate their activities. They needed mechanisms for cells to specialize, taking on different roles within the group. These innovations came later, built on the foundation eukaryogenesis established. For now, the crucial achievement is complete. Our lineage transformed from simple, small, energy-limited archaeal cells into large, complex, energy-rich eukaryotic cells capable of internal organization, active movement, predation, and sexual reproduction. Every animal, including humans, inherits this cellular architecture. Every cell in your body is a eukaryotic cell containing mitochondria, descended from that ancient bacterial merger, a nucleus organizing genetic information, an endomembrane system managing internal logistics, and a cytoskeleton providing structure and movement. The complexity visible in animals, tissues, organs, sophisticated behaviors, is built on the complexity established here, at the cellular level, 2 billion years ago. Our lineage now exists as single-celled eukaryotes, complex cells with mitochondria, nuclei, internal membrane systems, and cytoskeletons. These cells are larger, more energetically capable, and more structurally sophisticated than any prokaryote. For hundreds of millions of years, they diversify as single-celled organisms, exploring different ways to live in aquatic environments. This period sees the evolution of numerous protist lineages. The term protist simply means eukaryotes that aren't plants, animals, or fungi, and within this diversification, the specific adaptations emerge that will eventually enable multicellular life, including the lineage leading to animals. Early eukaryotes were predators. Unlike bacteria that absorb dissolved nutrients through their membranes, eukaryotes could engulf entire cells through phagocytosis. A eukaryotic cell extends protrusions of its membrane around a bacterium or smaller eukaryote, wraps completely around it, and internalizes it into a membrane-bound compartment called a food vacuole. Digestive enzymes break down the captured cell, and the nutrients are absorbed. This hunting lifestyle immediately creates a different selection pressure than those acting on absorptive bacteria. Success as a predator requires finding prey, capturing it efficiently, and avoiding becoming prey yourself. These demands drove the evolution of better sensory capabilities, proteins embedded in the membrane that detect chemical signals from potential prey or predators. They drove improvements in motility, the ability to move toward prey or away from threats, and they drove innovations in cell shape and behavior that made hunting more effective or escape more likely. Some eukaryotes evolved flagella, long whip-like appendages powered by molecular motors that propel the cell through water. Eukaryotic flagella are complex structures with a characteristic 9+2 arrangement of microtubules, 9 pairs forming a ring around 2 central singlets, all enclosed in a membrane extension from the cell surface. The microtubules slide against each other using motor proteins, creating wave-like beating motion that pushes water and moves the cell. Flagella allowed active swimming rather than drifting passively, enabling pursuit of prey and directed movement towards favorable conditions. Other eukaryotes evolved different solutions. Some developed cilia, shorter, more numerous hair-like projections covering the cell's surface that beat in coordinated waves, moving the cells or creating water currents that sweep prey towards the cell. Some became primarily sedentary, attaching to surfaces and extending sticky or adhesive projections to capture passing bacteria. Some abandoned predation entirely and involved photosynthesis, after engulfing photosynthetic bacteria in events similar to the mitochondrial merger, turning those bacteria into chloroplasts. That last point bears emphasis. Just as mitochondria arose from an archaeon engulfing a bacterium, chloroplasts arose from a eukaryote engulfing a photosynthetic bacterium. The lineages that acquired chloroplasts diverged into what we broadly call plants and various photosynthetic protists. This is not our lineage. Our ancestors remained non-photosynthetic, predatory cells. Plants and animals share a common eukaryotic ancestor from before the chloroplast acquisition, but after that point, their evolutionary paths separated. Plants solved the energy problem through photosynthesis. Our lineage continued relying on consuming other organisms. Within the predatory eukaryotes, one major group called Opisthecons emerged and diversified. Opisthecons includes animals, fungi, and several single-celled lineages. The defining feature is subtle. If they have a flagellum, it's single and positioned at the rear of the cell, pushing rather than pulling. This is a mere morphological detail, but it marks a real evolutionary grouping confirmed by genetic analyses. Our lineage belongs to Opisthecons, specifically to the branch within Opisthecons that led to animals. The closest living single-celled relatives of animals are cyanoflagellates, small predatory cells with a distinctive morphology. A cyanoflagellate has a single flagellum surrounding at its base by a collar of fine finger-like projections called microvilli. The flagellum beats, creating a water current that flows through the collar. Bacteria get trapped in the collar, stick to the microvilli, and are engulfed and digested. It's an efficient filter-feeding strategy that works well in bacteria-rich waters. Cyanoflagellates are important to our story for several reasons. First, they are single-celled cousins, the closest thing to what our pre-animal ancestors looked like. Second, they already possess some of the molecular tools animals later use for multicellularity. Cyanoflagellates have genes encoding proteins for cell adhesion, making cells stick together, and for cell signaling, allowing cells to communicate chemically. In cyanoflagellates, these proteins serve other functions or remain unused, but their presence indicates the molecular toolkit for multicellularity existed before animals evolved. Third, some cyanoflagellates form colonies. After a cell divides, the daughter cells sometimes stay attached rather than separating. A colony might contain a few cells or dozens, all genetically identical, all performing the same function or filter feeding, but physically connected. These colonies aren't multicellular organisms in the full sense. There's no specialization, no coordination, beyond physical attachment, but they demonstrate that staying together after division is possible and sometimes advantageous. Why would staying together benefit cells? Several advantages emerge even in simple colonies. A larger structure sinks more slowly than individual cells, staying suspended in the nutrient-rich upper water layers longer. A colony can create stronger water currents, pulling in prey from a larger area. A colony is harder for some predators to consume, too large to engulf whole, requiring predators to break it apart, which takes time and effort. These modest benefits can be enough for selection to favor cells that remain attached over cells that separate. The transition to true multicellularity requires more than just cells sticking together. It requires specialization, different cells taking on different roles. Specialization emerges through differential gene expression. All cells in a multicellular organism carry the same genome, but different cells activate different subsets of genes, producing different proteins and therefore different structures and functions. The mechanisms enabling differential gene expression existed in single-celled eukaryotes long before multicellularity. Even a solitary cell needs to regulate which genes are active in response to environmental conditions, turning on genes for digesting nutrients when food is available, activating stress response genes when conditions deteriorate, expressing different genes at different stages of the cell cycle. The regulatory networks controlling gene expressions were already sophisticated in single-cell ancestors. What changed in early multicellular organisms was using these regulatory systems in a spatial context. Cells in different positions within a colony experience different conditions. Cells on the outside face the external environment directly. Cells on the inside are shielded. Cells at the front of a moving colony encounter fresh water. Cells at the rear experience water already filtered by those in front. These differences in position create differences in signaling molecules the cells encounter, which trigger different regulatory responses, activating different genes. simple spatial patterns emerge automatically. In a spherical colony, outer cells might activate genes in response to detecting unfiltered water, while inner cells, not detecting that signal, leave those genes inactive. If outer cells then produce a signaling molecule that diffuse inwards, inner cells respond by activating different genes. Within a few cell divisions, the colony now has two cell types, outer cells expressing one set of genes and inner cells expressing a different set. This is the beginning of differentiation. Early differentiation was probably subtle and not particularly beneficial, but occasionally having different cell types provided advantages. Perhaps outer cells specialized in filter feeding while inner cells specialized in reproduction, protecting the next generation from environmental damage. Or perhaps some cells specialized in adhesion, producing extra sticky proteins that held the colony together more reliably. Any functional specialization that improved colony survival or reproduction would be favored by selection, gradually increasing the degree and complexity of differentiation. The evolution of extracellular matrix, materials that cells secrete around themselves, enhanced multicellularity substantially. Single-celled eukaryotes often secrete glycoproteins and other molecules that coat their surfaces, providing protection or helping them stick to substrates. Multicellular organisms elaborated these secretions into structured materials, filtering the spaces between cells. The extracellular matrix provided mechanical support, holding cells in specific geometric arrangements, and also served as a medium for diffusing signals between cells. Cell signaling systems became more sophisticated in multicellular contexts. Single cells already had receptors on their surfaces that detected external chemicals and triggered internal responses. In multicellular organisms, cells use similar receptors to detect signals from other cells in the same organism. A cell can release a signaling molecule that binds to receptors on nearby cells, triggering those cells to change their behavior, perhaps dividing or differentiating into a particular type or moving to a different location. These signaling systems allow coordination. Cells in different parts of a colony could respond to each other's states and adjust their own behavior accordingly. If cells at one end detected abundant food, they might signal cells at the other to stop dividing and focus on feeding. If cells in the center detected damage, they might signal outer cells to increase production of protective molecules. Coordination transformed a colony from a loose aggregate into a functional unit behaving coherently. The oxygen-rich environment, created by billions of years of cyanobacterial photosynthesis, played a crucial enabling role. Higher oxygen levels supported higher metabolic rates and allowed organisms to grow larger without their interiors suffocating. Early multicellular organisms were probably small, just dozens or hundreds of cells, but oxygen availability meant there was no fundamental barrier to growing larger. Oxygen diffuses through tissues, and organisms can remain aerobic as long as no cell is too far from an oxygen source. This becomes a constraint only at much larger sizes, eventually requiring circulatory systems. But for early simple multicellular organisms, ambient oxygen diffusion was sufficient. By about 800 million years ago, our lineage existed as small multicellular organisms, probably only a few hundred cells large, living in marine environments. These organisms had differentiated cell types, not many, perhaps only 2 or 3 distinct types, but enough to show functional specialization. They had extracellular matrix holding cells in defined arrangements. They had cell signaling systems allowing coordination. They could grow and reproduce, with some cells specializing in reproduction, while others specialized in feeding or structure. These weren't animals yet. They lacked tissues, organs, and the sophisticated organization that defines animals. But they had crossed the threshold from single-celled to multicellular. Every subsequent innovation in our lineage builds on this foundation. Groups of cells working together, specializing into different types, coordinating through signaling, and organized structurally through extracellular matrix. The path from here to recognizable animals involves elaborating these capabilities, adding more cell types, organizing those types into tissues, coordinating tissues into organs, and developing the specialized structures like nervous systems and muscles that allow complex behavior. That elaboration and the first true animals emerge in the next chapter. Our lineage now consists of small multicellular organisms with differentiated cell types, extracellular matrices, and cell signaling systems. They can coordinate behavior across hundreds of cells, but they lack the defining features that separate animals from other multicellular eukaryotes. What makes something an animal rather than just a colony of cooperating cells? Well, the answer lies in three interconnected innovations: specialized tissue organized into layers, coordinated movement through muscle-like cells, and nervous systems that allow rapid communication and response. These features emerged together between 800 and 600 million years ago, transforming our ancestors from simple multicellular organisms into the first true animals. The earliest animals were soft-bodied and left almost no fossils, but we can infer their structure from modern animals that retain ancient body plans and from rare fossil sites preserving soft tissue. The first animals were probably small, a few millimeters across, and radially symmetrical, like a cylinder or a sphere. They had an outer layer of cells facing the environment and an inner layer facing a central cavity. This two-layer organization, with an outer ectoderm and an inner endoderm, separated by a jelly-like material, created the basic animal architecture. The critical innovation was the gut, a central cavity opening to the outside where digestion occurred externally to cells. Single-celled eukaryotes engulf food and digest it inside inner membrane-bound compartments. Early multicellular colonies probably did the same, with each cell feeding itself, but a shared gut changed everything. Cells lining the inner cavity secreted digestive enzymes into the space, breaking down food items too large for any single cell to engulf. The digested nutrients were then absorbed by the lining cells and distributed to other cells in the organism. This innovation enabled larger size and specialization. Cells on the outer surface no longer needed to feed themselves directly. They received nutrients from inner cells. This freed outer cells to specialize in protection, sensing the environment, or capturing prey. Inner cells specialized in digestion and nutrient distribution. The organism became more than a sum of its cells, because different cells performed different essential functions, creating interdependence. Coordinated movement required contractile cells, the evolutionary precursors to muscles. Early versions were probably simple epithelial cells that could change shape by contracting protein fibers running through their interior. Modern animals use actin and myosin proteins that slide past each other, generating force. These proteins existed in single-celled ancestors where they enabled cell crawling and shape changes. In early animals, cells began specializing in contraction, organizing their actin and myosin into aligned bundles that generated stronger, more directed forces. When contractile cells on one side of the organism contracted simultaneously, the body bent toward that side. When cells around the circumference contracted together, the body constricted. These simple movements, bending, constricting, extending, allowed the organism to reorient itself toward food or away from harmful conditions, to pull itself along a surface, or to change shape to capture prey. Movement was slow and imprecise compared to modern animals, but it was deliberate and responsive rather than passive drifting. Coordination required communication faster than diffusing chemicals could provide. If detecting food on the left side required minutes for a chemical signal to diffuse across the organism to trigger contractile cells on the right side, the opportunity would probably pass. The solution was nerve cells, elongated cells specialized for rapid electrical signaling. The basic mechanism exploits the fact that cell membranes maintain voltage differences through ion pumps that push sodium ions out and keep potassium ions in. When a nerve cell receives a stimulus, a chemical signal from a sensory cell detecting food, for example, channels in the membrane open, allowing sodium to rush back in. This voltage change triggers adjacent channels to open, creating a wave of depolarization racing along the nerve cell's length at speeds measured in meters per second rather than millimeters per second of diffusion. Where the nerve cell contacts a contractile cell, the electrical signal triggers release of chemical neurotransmitters that bind to receptors on the contractile cell, causing it to contract. A network of nerve cells connecting sensory cells to contractile cells throughout the body allowed coordinated responses within fractions of a second. An organism detecting food on its left side could bend left almost immediately, tracking the source while it remained nearby. These three innovations, layered tissue organization with a gut, contractile cells enabling movement, and nerve cells enabling coordination, define animals. Every animal, from sponges to humans, possess some version of these features. Sponges are the exception that proves the rule. They have differentiated cell types and coordinated feeding currents, but they lack true nerves and muscles and cannot move as whole organisms. Most biologists consider sponges animals, but the earliest branching animals, representing the baseline from which everything else elaborated. Our lineage moved past the sponge-grade organization toward more complex animals called metazoans, animals with true tissues, nerves, and muscles. The earliest metazoans lived between 650 and 600 million years ago, during a period of dramatic environmental change. Earth had just emerged from a severe glaciation period, where ice sheets extended nearly to the equator. As the planet warmed and ice retreated, ocean chemistry changed, oxygen levels rose further, and new ecological opportunities opened. These early metazoans resembled modern cnidarians. They had radial symmetry, meaning their bodies were organized around a central axis, like spokes on a wheel. They had tentacles surrounding a mouth opening into a central gut cavity. The tentacles, armed with specialized stinging cells, captured small prey and pushed them into the mouth. Nerve cells form a loose network throughout the body, allowing coordinated tentacle movements and body contractions. The Ediacaran period, from 635 to 541 million years ago, saw these early animals diversify into a variety of body forms, preserved as fossils in a few exceptional sites. The fossils show disc-shaped organisms, frond-like organisms anchored to the seafloor, and segmentic worm-like organisms. Many of these are difficult to interpret. They don't fit cleanly into modern animal groups, and some might represent extinct experimental body plans that left no descendants. What's clear is that by 550 million years ago, animals had spread across the sea floor in shallow marine environments. They remained relatively simple. Soft-bodied, small, probably slow-moving. No hard shells, no skeletons, or complex organs. But they were unquestionably animals. Multicellular organisms with differentiated tissues, guts, nerves, and muscles, actively moving and feeding on other organisms or filtering particles from water. The constraint limiting these early animals was predation, or rather, the lack of it. Without significant predators, there was little pressure to evolve defenses like shells or escape behaviors like rapid swimming. Without active predators, there was less advantage to evolving sophisticated sensory systems or powerful muscles. The Ediacaran fauna existed in a relatively peaceful world where competition for space and food mattered more than avoiding being eaten. This changed at the Ediacaran-Cambrian boundary around 541 million years ago. The fossil record shows a sudden shift. Ediacaran-style organisms disappear, and new animals forms with hard shells, mineralized skeletons, and evidence of predation appear. This transition marks the beginning of the Cambrian explosion. But before we get there, we need to identify where our specific lineage was during the Ediacaran. Our ancestors were probably small, soft-bodied, worm-like organisms living on or in seafloor sediments. They weren't the large fawn-like fossils that dominate some Ediacaran sites. They were bilaterans, animals with bilateral symmetry, meaning their bodies had distinct left and right sides, a front and back, and a top and bottom. This body plan, which seemed obvious for any mobile animal today, was an innovation during this period. Bilateral symmetry makes sense for directed movement. A radially symmetric animal, like a jellyfish, moves vertically or drifts with currents. It has no preferred horizontal direction. A bilaterally symmetrical animal moves forward with sensory organs concentrated at the front to detect what it's approaching, a mouth positioned to consume what it encounters, and propulsive structures pushing from behind. This organization creates a head-tail axis and a dorsal-ventricle axis, fundamentally restructuring the body compared to radial ancestors. The genetic mechanisms enabling bilateral symmetry involve Hox genes, regulatory genes that specify position along the head-tail axis. These genes existed in earlier animals, but became elaborated in bilaterans into a coordinated system that divided the body into distinct regions, each with different structures. Hox genes act as master switches, activating different sets of downstream genes in different body regions. The front expresses certain Hox genes and develops head structures. The middle expresses different combinations and develops trunk structures. The rear develops tail structures. Early bilaterans were likely small and worm-like because this body plan suits burrowing. As animals evolved to exploit sediments for food and shelter, bilateral symmetry and elongated bodies became advantageous. An organism could push its front end forward through sediment, using muscles to generate sequential waves of contraction traveling from head to tail, propelling itself efficiently through a dense medium. Our specific ancestors during the late Ediacaran were probably acoelomates, bilaterans lacking a body cavity between the gut and outer body wall. Their insides were solid tissue except for the central gut cavity. This limited their size because nutrients and oxygen had to diffuse through tissue from the gut and outer surface to reach internal cells, but it was mechanically simpler and sufficient for small organisms. By the end of the Ediacaran, around 541 million years ago, our lineage consisted of small, bilaterally symmetrical, soft-bodied worms a few millimeters to perhaps a centimeter long. They had a through gut, a mouth at one end, and an anus at the other, allowing one-way flow of food and more efficient digestion than the single-opening guts of cnidarians. They had longitudinal and circular muscles, allowing sophisticated crawling and burrowing. They had a concentration of nerve cells at the front end, forming a simple brain, coordinating sensory input and motor output. They likely had simple, light-sensitive cells detecting direction and intensity but not forming images. These animals weren't impressive by modern standards. They'd be pretty easy to overlook in a modern tidal pool, but they possessed the fundamental bilateran body plan that every animal in our lineage since has elaborated. The head-tail axis, the left-right symmetry, the dorsal-ventral organization, the through gut, the anterior nerve concentration, these features, established during the Ediacaran, remains visible in every vertebrate, including humans. The stage was now set for the most dramatic diversification in animal history. Oxygen levels continued rising, approaching modern concentrations. Ecological relationships intensified as animals evolved to prey on each other more effectively. And crucially, animals evolved biomineralization, the ability to construct hard structures from calcium carbonate, calcium phosphate, and silica. Hard parts enabled new defenses, new modes of predation, new body support systems, and crucially for paleontologists, a fossil record that suddenly becomes rich and detailed. The explosion of animal diversity preserved in that fossil record and our lineage's role in it is the next chapter. At 541 million years ago, the fossil record transforms. For 3 billion years, life left mostly microscopic traces, bacterial mats, single cells, and the occasional soft-bodied impression. Suddenly, within a window of perhaps 20 million years, fossils appear showing true animals with hard shells, complex eyes, jointed appendages, and sophisticated predatory structures. This is the Cambrian explosion. Not an explosion of life itself, which had been diversifying for millions of years, but an explosion of preservation as animals evolved mineralized body parts that fossilize readily. More importantly for our story, this is where the basic body plans of all major animal groups, including our own phylum Chordata, first appear in the fossil record. The trigger for this rapid diversification was an evolutionary arms race. Once one lineage evolved hard protective shells from calcium carbonate, any predator that couldn't break through those shells starved, while competitors that evolved stronger jaws or more powerful crushing appendages thrived. Prey responded by thickening shells or adding spines. Predators responded with more sophisticated hunting techniques, better sensory systems to detect hidden prey, and faster locomotion to chase down escapees. Each innovation on one side created selective pressure for counter-innovations on the other, driving rapid evolution across all animal lineages simultaneously. The chemistry of biomineralization, building hard structures from minerals, wasn't entirely new. Some ediacaran animals had produced small mineralized elements, and prokaryotes had been precipitating minerals for billions of years. But Cambrian animals systematized and controlled the process. Specialized cells secreted proteins that formed a scaffold, then deposited calcium carbonate or calcium phosphate crystals onto that scaffold in organized layers. The result was shells and skeletons with specific shapes and properties, grown and maintained by the organism rather than simply precipitated from surrounding water. Hard parts enabled larger size. Soft-bodied animals are limited by structural integrity. They collapse under their own weight beyond a certain size, especially in air or when moving against resistance. A mineralized skeleton, whether external like a shell or internal like bone, provides structural support, allowing much larger bodies. Larger size provides advantages in complexion and predation, but requires more sophisticated systems for circulating nutrients and oxygen to distant tissues. Our lineage during the Cambrian was not among the dominant or most visually striking animals. Arthropods, animals with jointed external skeletons and segmented bodies, including trilobites, dominated Cambrian seas. Mollusks with hard shells were abundant. Various worm-like animals with plates, spines, or tubes thrived. Our ancestors remained small, soft-bodied, and superficially unremarkable. They're preserved in only a few exceptional fossil sites where soft tissue were rapidly buried and mineralized before decaying. The most famous site is the Burgess Shale in Canada, where 508 million-year-old fossils preserve extraordinary detail, including soft tissues, guts, and even traces of eyes and nervous systems. Among the bizarre Cambrian fauna, five-eyed predators walking cactus-like animals, bizarre filter feeders, are several small organisms that represent our lineage's position during this era. They are early chordates, animals possessing a notochord, a flexible rod running along the back that provides structural support. The notochord is the defining feature of our phylum. It's not yet a backbone, just a stiff but flexible rod made of turgid cells surrounded by a tough sheath. But it's positioned dorsally, along the back, and it serves the same basic function that the backbone will later serve, providing an internal skeleton that muscles can pull against for efficient swimming. Animals with notochords could undulate their bodies in coordinating waves, each muscle segment pulling against the notochord's resistance, generating thrust more efficiently than worms that lacked any internal support structure. Early chordates also had segmented muscles, repeated blocks of muscle tissue running along both sides of the body from head to tail. This segmentation, called myomeres, allowed sequel contraction, creating smooth swimming motion. When muscles on the left contracted from head to tail in a wave, while muscles on the right relaxed, the body curved left. When the pattern reversed, the body curved right. Alternating these patterns rapidly generated a side-to-side swimming motion, propelling the animal forward. The earliest well-preserved chordate from the Cambrian is Pikaia, a small animal about 5 centimeters long, found in the Burgess Shale. Pikaia had a clear notochord, segmented muscles, and a distinct head with simple sensory organisms. It swam above the sea floor, probably filter feeding by drawing water through its mouth and filtering out small particles. It was not a fast or powerful swimmer, but it was more efficient than most contemporary worms. Related to Pikaia, but showing additional features are animals like Haikuixis and Mailongkungmingjua from slightly older Chinese fossil deposits around 518 million years ago. These animals, just 2 to 3 centimeters long, show not only a notochord and segmented muscles, but also possible evidence of cartilaginous elements supporting the gills and perhaps stiffening the head region. They represent steps towards vertebrates, animals with a backbone made of repeated skeletal elements called vertebrae. The importance of an internal skeleton cannot be overstated for our lineage's later success. External skeletons, like those of arthropods, provide excellent protection and support, but create constraints. To grow, an arthropod must molt, shed its entire external skeleton, grow rapidly while soft and vulnerable, then harden a new, larger skeleton. This limits maximum size because the period of vulnerability increases with body size. Internal skeletons grow continuously without molting, allowing much larger ultimate size without extended vulnerability. Another chordate feature visible in these Cambrian animals is the postanal tail. The body extends past the anus, providing additional surface area for muscles and propulsion. Most invertebrates end at the anus. Chordates have a muscular tail. This seems minor, but matters enormously for swimming efficiency. The tail provides extra thrust without adding digestive organs or other metabolically expensive tissue, allowing faster swimming for lower energy cost. Early chordates also show a dorsal nerve cord, a hollow tube running along the back above the notochord, formed from ectoderm that rolled into a tube during development. This contrasts with invertebrates, which typically have solid ventral nerve cords running along the belly. The dorsal position, hollow structure, and formation mechanism of the chordate nerve cord indicate it evolved independently from invertebrate nerve cords, representing a distinct solution to the problem of organizing nervous system anatomy. By the Middle Cambrian, around 510 million years ago, our lineage consisted of small swimming chordates with notochords, segmented muscles, dorso-hollow nerve cords, pharyngeal slits used in filter feeding, postanal tails, and increasingly sophisticated heads with concentrated sensory organs. These animals were not dominant predators or ecosystem engineers. They were modest participants in Cambrian ecosystems, probably eaten regularly by arthropods and other predators occupying a middle tier in the food web. But the chordate body plan had advantages that would compound over time. The notochord provided efficient swimming. The dorsal nerve cord could expand and elaborate without interfering with gut function below it. The internal support structure could be modified and added to without requiring complete redesign. The segmented muscle plan allowed fine control of swimming motion. These features created a platform for later innovations. One critical evolutionary transition during the late Cambrian was the development of true vertebrae, repeated skeletal elements surrounding and eventually replacing the notochord. The earliest vertebrae were probably small cartilaginous arches protecting the nerve cord, appearing in addition to the notochord. Over time, these arches expanded, became more substantial, and began providing structural support themselves. The notochord persisted between the vertebrae, eventually becoming reduced to the shock-absorbing discs between vertebrae in modern vertebrates. Vertebrae provided several advantages over a simple notochord. They created a more rigid structure when needed while allowing flexibility between adjacent vertebrae. They protected the nerve cord from damage more effectively than soft tissue alone. They provided attachment points for muscles, improving swimming efficiency, and they could be modified along the body's length, larger in some regions, smaller in others, with different projections or shapes, allowing regional specialization of the body plan. The head region underwent particular elaboration. Early chordates had simple heads with light-sensitive cells and chemical sensors. By the late Cambrian, heads were developing cartilaginous structures supporting larger, more sophisticated sense organs. The front of the nerve cord expanded into a more defined brain, with distinct regions processing different types of sensory information. Eyes capable of forming images evolved, using similar optical principles to arthropod eyes, but with different developmental origins. Eyes transformed ecological interactions. An animal that can see predators approaching from a distance can flee earlier, improving survival. An animal that can see prey can hunt more effectively, improving feeding success. Vision created strong selective pressure for faster swimming, both to chase prey and escape predators, which drove improvements in muscle organization, fin structure, and body streamlining. By the end of the Cambrian, around 485 million years ago, our lineage existed as small vertebrates, a few centimeters long, with cartilaginous skeletal elements including vertebrae and skull components, segmented muscles, paired fins beginning to develop from lateral folds of tissue, image-forming eyes, and a three-part brain. They were active swimmers, probably hunting small invertebrates and fleeing from larger predators. They had moved from filter feeding to predatory feeding, requiring more sophisticated sensory systems and faster, more controlled movement. These early vertebrates set the stage for the next major transition, the evolution of jaws. For the entire Cambrian and into the following Ordovician period, all vertebrates were jawless, feeding either by filter feeding or by sucking up small prey items whole. But hidden within the architecture of the pharyngeal region, the gill-bearing area behind the head, were skeletal elements that could be modified into jaws. That modification, when it occurred, transformed vertebrates from minor participants in marine ecosystems into dominant predators. Our lineage's rise to ecological prominence begins with that innovation, which we'll explore in the next chapter. For now, the critical achievement is complete. Our ancestors passed through the Cambrian as small but increasingly sophisticated chordates, developing the vertebrate body plan that every animal in our lineage since has inherited. The notochord became a backbone. The dorsal nerve cord became a spinal cord topped by an elaborating brain. Simple sense organs became sophisticated eyes and other sensory systems. A modest filter feeder became an active predatory swimmer. Every structural feature that makes vertebrates distinctive, including the features that make humans distinctive among vertebrates, traces back to innovations established during this explosive period of animal evolution. Our lineage now exists as small vertebrates with cartilaginous skeletons, segmented muscles, paired fins, and sophisticated sensory systems. These early vertebrates swim actively through Ordovician seas around 480 million years ago, hunting small prey and evading predators. But they share a critical limitation with all vertebrates for the first 50 million years of vertebrate history. They lack jaws. Every early vertebrate feeds either by filter feeding, straining small particles from water, or by sucking up tiny prey whole. This constraint limits what they can eat, how large they can grow, and their role in ecosystems. The evolution of jaws, one of the most consequential innovations in our lineage's history, transforms vertebrates from peripheral players into dominant predators. Jawless vertebrates, called agnathoms, diversified considerably during the Ordovician and Silurian periods. Some remained small and eel-like, others evolved heavy armor, bony plates covering the head and front body, probably as defense against the giant predatory arthropods and cephalopods that dominated these ancient seas. Some developed elaborate head shields with sensory grooves, suggesting they hunted by detecting electrical fields produced by prey muscles, much like modern sharks. But all fed through a simple opening at the front of the head, lacking any mechanism to bite, tear, or process large food items. The constraint wasn't lack of food. Ordovician and Silurian seas teemed with invertebrates, algae, and other vertebrates, but lack of access to much of it. A heavily armored trilobite was inaccessible to a jawless fish that couldn't crack the shell. A fast-swimming cephalopod could escape a predator that needed to approach close enough to suck it in. Large food items were impossible to consume. The vertebrate that could overcome this constraint would access vastly more food resources than competitors. The solution came from an unexpected source: gill supports. All fish have pharyngeal arches, paired cartilaginous or bony bars that support the gills in the throat region. Water enters through the mouth, passes over the gills where oxygen is extracted, and exits through gill slits supported by these arches. Early vertebrates had 6 or 7 pairs of pharyngeal arches, all serving this respiratory function. The first pair sat just behind the mouth opening, the second pair behind that, and so on. Around 430 million years ago, in the Silurian period, some vertebrate lineages began modifying the first pair of pharyngeal arches. Instead of supporting gills, these arches expanded forward and developed hinges, creating a mechanism that could open and close around the mouth. The upper arch attached to the skull and formed an upper jaw. The lower arch became a lower jaw, hinged to the skull at the back and mobile at the front. The second pair of pharyngeal arches, just behind the first, modified to support and stabilize the jaw joint. This transformation, serial homology, where structures that originally repeated along the body became modified for different functions, is a recurring pattern in evolution. The pharyngeal arches didn't appear suddenly as jaws. They were already present, performing a different function, with existing developmental programs controlling their growth and shape. Evolution modified those existing programs slightly, redirecting development, creating new structures from old parts. The earliest jawed vertebrates, called placoderms, appeared around 420 million years ago in the late Silurian and radiated dramatically through the Denovian period. Placoderms varied enormously in size and form. Some remained small, a few centimeters long. Others grew to massive sizes. Dunkleosteus, perhaps the most famous placoderm, reached 6 to 9 meters in length, with blade-like bony jaw plates capable of shearing through prey with tremendous force. Placoderms dominated marine and freshwater environments for over 50 million years. But placoderms are not our ancestors. They represent an early experiment in jawed vertebrate design that eventually went extinct around 360 million years ago, outcompeted by other jawed fishes. Our lineage during the Denovian belonged to a different group of jawed fishes that split from placoderms early in jaw evolution. The gnathostomes, which include all modern jawed vertebrates, sharks, bony fish, and tetrapods including humans. The advantage jaws provided was access to new food sources through 3 mechanisms: biting, cutting, and holding. A predator with jaws could bite chunks from prey too large to swallow whole. It could cut through shells and armor, protecting soft tissue underneath. It could hold struggling prey while positioning it for swallowing or while tearing it apart. Each capability opened ecological niches previously unavailable to jawless vertebrates. Jaws also enabled suction feeding to become more powerful and controlled. Even modern jawed fish that don't bite often use rapid jaw expansion to create strong suction, pulling prey and water into the mouth. The ability to precisely control mouth volume and shape through jaw movements made suction feeding more effective than the simple pumping mechanisms jawless fish used. Teeth evolved shortly after jaws, initially as modified scales. Fish bodies were covered with small scales made of dentin, the same material that forms teeth, covered with enamel-like substances. Scales near the mouth enlarged and became more pointed, creating structures useful for gripping prey. These prototeeth were not deeply anchored or replaced regularly like modern teeth, but they provided enough grip to prevent prey from escaping once captured. Concurrent with jaw evolution, paired fins became more sophisticated. Early vertebrates had simple lateral folds or fin-like projections from the body, but jawed vertebrates developed distinct pectoral fins behind the head and pelvic fins further back, both supported by internal cartilaginous or bony elements. These paired fins provided stability and maneuverability. A swimming fish generates forward thrust by undulating its body and tail, but this creates instability. The fish tends to roll, pitch, or yaw. Paired fins act as hydrofoils, counteract these unwanted movements, allowing faster, more controlled swimming. Better swimming, combined with predatory jaws, created an evolutionary feedback loop. Effective predators needed to catch prey, requiring speed and maneuverability, driving improvements in fins, muscle organization, and body streamlining. Effective prey needed to escape, requiring the same improvements. The result was an arms race where both predators and prey in our lineage's branch became progressively faster, more agile, and more efficient swimmers. Our specific ancestors during the Devonian belonged to Osteichthyans, or bony fish. This group split from cartilaginous fish early in gnathostome evolution. The defining features is bone, not just cartilage, but true mineralized bone made of calcium phosphate deposited in an organic matrix. Bone is harder and more rigid than cartilage, providing better support and protection. It's also metabolically active tissue that can be remodeled and repaired throughout life, unlike cartilage, which grows slowly and doesn't heal well. Bony fish also developed swim bladders, or gas-filled sacs derived from outpocketings of the gut. By adjusting the gas volume in the swim bladder, a fish can control its buoyancy, maintaining neutral buoyancy at a given depth without constantly swimming to avoid sinking. This energy saving is substantial. A fish with a swim bladder uses far less energy maintaining position than a shark, which must swim constantly or sink. This innovation may seem minor, but it allowed bony fish to occupy different ecological niches, hovering in place to ambush prey or hiding in vegetation without expending constant energy. The swim bladder is evolutionarily crucial for another reason. It's homologous to lungs. The same developmental outpocking that becomes a gas-filled buoyancy organ in most bony fish became a gas exchange organ in others. In our lineage, this outpocketing becomes lungs, enabling air breathing and eventually allowing vertebrates to move onto land. But that's several chapters ahead. Devonian bony fish diversified into two major groups, ray-finned fish and lobe-finned fish. Ray-finned fish have fins supporting by thin bony rays radiating from the body, with muscles for moving the fins located primarily in the body wall. They're efficient swimmers with highly maneuverable fins, and they became the most successful vertebrate group in terms of species diversity. Today, ray-finned fish account for roughly half of all vertebrate species. But our lineage is not with ray-finned fish. We descend from lobe-finned fish, Sarcopterygians, a group that took a different structural approach. Lobe-finned fish have fleshy, muscular fins supported by a core of bones rather than thin rays. The bones within the fins are homologous to bones in your arm, the humerus, radius, and ulna, and smaller bones in the wrist and hand. The muscles that move the fin are within the fin itself, attached to these bones, allowing more powerful and varied movements than ray-finned fish. In the water, these muscular fins provided advantages in certain environments, particularly shallow waters with complex bottom structures, where pushing against substrate or maneuvering through vegetation benefited from fins that could exert force in multiple directions. But the real significance of lobe-finned anatomy becomes apparent only later, when descendants of these fish move onto land. The bones and muscles already present in lobe-finned fish become, with modification, the bones and muscles of tetrapod limbs. By the late Devonian, around 380 million years ago, our ancestors were lobe-finned fish with jaws and teeth, sophisticated paired fins containing bone and muscle, a bony internal skeleton including a backbone of distinct vertebrae, a swim bladder that could be used for buoyancy and potentially for supplementing gill respiration with air breathing, and a well-developed brain with large eyes and other sensory organs. They ranged from small, perhaps 10 centimeters, to medium-sized, perhaps 50 centimeters or more. These fish lived primarily in freshwater environments, rivers, lakes, and swamps. Freshwater poses challenges saltwater doesn't. Oxygen levels fluctuate more dramatically, temperatures vary more widely, water levels change seasonally, sometimes leaving pools isolated, and habitats can become stagnant. These challenges selected for physiological flexibility, particularly the ability to tolerate low oxygen by breathing air at the surface using the swim bladder as a primitive lung. The late Devonian environment was setting the stage for the next major transition. Extensive shallow water environments, rich in vegetation, developed along continental margins. Invertebrates, including early insects, had already colonized land. Plants had established complex ecosystems in wetland areas. Food resources existed on land in the form of plants and invertebrates, but no vertebrate could access them. The vertebrate lineage that could move from water onto land, even briefly, would access an entirely new realm of resources. That transition requires modifications to fins, breathing, skeleton, and sensory systems, changes that would transform lobe-finned fish into tetrapods, the four-legged vertebrates that eventually give rise to amphibians, reptiles, mammals, and humans. But the foundation was already in place. The bony skeleton provided structural support that cartilage couldn't match. The muscular lobed fins contained the bone structure that could be modified into walking limbs. The swim bladder was pre-adapted to functioning as a lung. The sophisticated sensory systems, particularly vision and smell, could function in air with minor modifications. Our lineage stood at the water's edge, literally and evolutionarily. Everything that came before, the origin of animals, the development of bilateral symmetry, the evolution of chordates, the appearance of jaws, the elaboration of bony skeletons and muscular fins, had created a vertebrate capable of exploiting terrestrial environments. The transition from fish to tetrapod, one of the most studied and best documented transitions in the fossil record, is next. Our lineage now exists as lobe-finned fish living in shallow freshwater environments during the late Devonian, around 380 million years ago. These fish have muscular fins supported by internal bones, functional lungs derived from swim bladders, and the ability to tolerate low oxygen conditions by breathing air at the surface. They inhabit river deltas, swamps, and seasonal pools where water levels fluctuate dramatically and oxygen depletion is common. The transition from these fish to four-legged land animals, or tetrapods, happen in this challenging aquatic environment, not through fish suddenly deciding to explore land, but through incremental adaptations to living in shallow, plant-choked, oxygen-poor water. The late Devonian climate was warm and seasonal. During wet seasons, rivers and swamps flooded, connecting waterways and creating expansive shallow habitats. During dry seasons, water level dropped, breaking continuous waterways into isolated pools. Fish trapped in these pools faced two threats: oxygen depletion, as decaying vegetation consumed dissolved oxygen, and complete drying if the pool evaporated. Any adaptation that helped fish survive these conditions, like better air breathing, the ability to move overland to find deeper water, or tolerance of partial desiccation, provided significant survival advantages. The earliest modifications involved the fins. Lobe-fin fish already had bones inside their fins, homologous to tetrapod limb bones, with one large bone near the body, two parallel bones further out, and smaller bones at the fin tip. These bones were surrounded by muscle and covered in scaled skin. To make these fins more useful for pushing through dense vegetation or supporting weight on the bottom substrate, the bones became more robust and the joints between them became more flexible and better defined. Fossil intermediaries preserve this transition beautifully. Eustenoperon, from about 385 million years ago, was a fully aquatic lobe-fin fish with muscular fins, but a streamlined body suited for swimming. Its fins could push against objects, but couldn't support the body weight in air. Panderichthys, from 380 million years ago, shows a flatter body profile, eyes positioned on top of the head rather than on the side, and fin bones that are thicker and more limb-like. This animal probably spent time resting on the bottom of shallow water pools, using its fins to prop itself up and raise its head above the waterline to breathe. The most famous intermediate is Tiktaalik, discovered in Arctic Canada in rocks 375 million years old. Tiktaalik was about 2 to 3 meters long, with a crocodile-like head, a flexible neck, which is unusual for fish, which typically have skulls rigidly connected to shoulder girdles, and fins that had clearly become more limb-like. The fin bones were strong enough to support the animal's weight. The wrist-like joints could bend, allowing the fin to push backwards against substrate, moving the animal forward. The ribs were enlarged, possibly helping support the body when partially out of water, and definitely providing support for lungs that had become more elaborate than simple swim bladders. Tiktaalik probably lived in shallow streams and swamps, hunting smaller fish and possibly emerging partially onto mud banks or logs. It wasn't walking in the sense tetrapods walk. It was still primarily aquatic, but it could use its fins to prop itself up, to push through dense vegetation, and to move short distances across obstacles or between isolated pools. These behaviors, originally adaptations to aquatic environments, pre-adapted the lineage for terrestrial locomotion. The next clear fossil is Acanthostega, from 365 million years ago in Greenland. Acanthostega had 8 digits on each limb, more than the 5 standard in later tetrapods. Its limbs had well-defined wrists and ankles, with multiple bones allowing complex movements, but crucially, the limb proportions and joint structure show it was still primarily aquatic. The limbs were better suited for paddling through water than walking on land. The ribs were too short to support the body effectively in air. The tail was deep and paddle-like, perfect for swimming. This reveals something important. Digits and limb-like structures evolved in the water for aquatic uses before vertebrates regularly moved onto land. The digits probably helped with pushing through vegetation, gripping substrate in fast-moving water, or maneuvering in complex three-dimensional aquatic environments. Only later were these structures co-opted for walking. Breathing was already partially solved. Lobe-fin fish ancestors had functional lungs, but they also retained gills for extracting oxygen from water. The transition to tetrapods involved lungs becoming larger and more efficient, while gills gradually reduced. Early tetrapods, like Acanthostega, retained both systems, probably breathing primarily through lungs, but still using gills when submerged. Later tetrapods lost gills entirely in the adult stage, though some retained them during larval development. The circulatory system required modification. Fish have a two-chambered heart that pumps blood to the gills where it picks up oxygen, then directly to the body, then back to the heart. When lungs replace gills as the primary gas exchange organ, blood must be pumped to lungs, return to the heart, then be pumped to the body. This requires separating the heart into more chambers to prevent oxygen-poor blood returning from the body from mixing with oxygen-rich blood returning from the lungs. Early tetrapods developed partially divided hearts with 3 chambers, 2 atria and 1 ventricle with incomplete separation. This is less efficient than the fully 4-chambered hearts of mammals, but adequate for animals with low metabolic rates. Sensory systems adapted to air. Eyes that evolved underwater work poorly in air without modification because water and air have different refractive indices. The cornea, which provides most of the eye's focusing power in air, provides almost no focusing power underwater. Early tetrapods developed modifications to the cornea and lens to restore proper focusing in air. The lateral line system, sensory organs along fish bodies that detect water pressure and movement, become less useful out of water and eventually reduced or lost in fully terrestrial forms. Hearing required the evolution of a middle ear. Sound travels well through water, and fish detect it through bone conducting vibrations directly to the inner ear. Sound travels through air differently, requiring a mechanical system to amplify the small pressure waves hitting the outer ear and transmit them into the inner ear fluid. Early tetrapods modified one of their gill arch bones, the hypomandibular, which in fish brace the jaw against the skull, into the stapes, the first bone of the tetrapod middle ear. This bone bridged from the eardrum to the inner ear, conducting airborne sound inward. Later tetrapods added additional middle ear bones refined from other skull elements. Skin became a major challenge. Fish skin is covered with scales and kept moist by surrounding water. Tetrapod skin exposed to air desiccates rapidly unless protected. Early tetrapods retained scales, which provided some protection, but also developed mucus glands that secreted moisture, keeping the skin surface damp. Modern amphibians still use this solution, maintaining thin, moist, scaleless skin and remaining tied to human environments. Later tetrapods, reptiles and their descendants, evolved thicker skin with waterproof keratin layers, completely solving the desiccation problem. By about 360 million years ago, at the end of the Devonian period, our lineage consisted of animals that were recognizably tetrapods, 4-legged vertebrates with distinct limbs rather than fins, with digits and a neck allowing independent head movement, and with lungs as a primary breathing organ, and with various adaptations allowing them to spend significant time out of water. But they weren't fully terrestrial. They still lived in and around water, probably spending most of their time partially submerged, returning to water to reproduce, and hunting primarily in aquatic or semi-aquatic environments. These early tetrapods diversified rapidly in the Carboniferous period that followed. Some remained semi-aquatic. Others became increasingly terrestrial, developing stronger limbs, more efficient lungs, and better desiccation resistance. The Carboniferous climate was warm and wet, with vast swamp forests covering much of the land. These forests provided abundant food. Insects had diversified dramatically, including giant dragonflies and millipedes, and abundant shelter from the few predators that had made the transition to land. One critical innovation appeared during the Carboniferous, the amniotic egg. Early tetrapods, like modern amphibians, laid their eggs in water. The eggs were similar to fish eggs, small, jelly-covered, permeable to water, requiring an aquatic environment to prevent drying out. This tied reproduction to water bodies, even if adults could live on land. The amniotic egg, which we'll explore in detail in the next chapter, enclosed the embryo in a shell with internal fluid-filled membranes, creating a self-contained aquatic environment the embryo could develop in anywhere, regardless of external conditions. But before this innovation, our ancestors existed as early tetrapods, exploring the margins between water and land. They retained many fish-like features: scales, dependence on moist environments, aquatic larvae, relatively simple limbs. They were opportunistic predators, eating whatever they could catch. Fish when in water, invertebrates when on land. They varied in size from perhaps 20 centimeters to over a meter in length. They represented dozens of different lineages, experimenting with different solutions to terrestrial living. By the end of the Carboniferous, around 300 million years ago, our specific lineage had become fully terrestrial in the adult stage, with limbs adapted for walking rather than swimming, skin resistant to desiccation, and sensory systems optimized for air rather than water. But one constraint remained: reproduction still required returning to water to lay eggs. The evolution of the amniotic egg, which freed reproduction from water and opened the vast interior continental regions to colonization, marks the next transformative step. That innovation defines a group called amniotes, which include all reptiles, birds, and mammals, every terrestrial vertebrate that doesn't need to return to water to breed. Our lineage from that point forward is amniote, and understanding what that means is essential to understanding the path leading to mammals and eventually humans. Our lineage now consists of fully terrestrial tetrapods living in Carboniferous swamp forests around 320 million years ago. These animals walk on strong limbs, breathe air efficiently, and have skin resistant to water loss. They hunt the enormous insects thriving in oxygen-rich forests, dragonflies with 70-centimeter wingspans, millipedes a meter long. But one critical constraint remains: they must return to water to lay their eggs. Their eggs are like fish eggs, small, jelly-coated, and permeable. On land, they dry out within minutes. This ties reproduction to ponds and streams, limiting where these animals can live and making them vulnerable during breeding. The innovation that breaks this constraint, the amniotic egg, opens the vast continental interior to vertebrate colonization for the first time. The amniotic egg is essentially a portable aquarium. The embryo develops inside a fluid-filled sac called the amnion, floating in its own private water environment regardless of external conditions. A second membrane, the allantois, spreads beneath the shell and serves as both a respiratory surface, with blood vessels bringing embryonic blood close to the porous shell where oxygen enters and carbon dioxide exits, and a waste storage bag where toxic nitrogen compounds accumulate safely until hatching. The yolk sac provides all nutrients needed for complete development. The shell itself is porous enough for gas exchange, but structured to minimize water loss. This system solves three problems simultaneously: desiccation, respiration, and waste disposal. The embryo develops from single cell to hatching entirely within the egg, never requiring external water. The first amniotes could reproduce anywhere: in dry uplands, deep forests, seasonal environments where water existed only part of the year, continental interiors, previously inaccessible, suddenly opened to colonization. By 312 million years ago, amniotes had split into two major lineages, distinguished by skull openings behind the eye. One lineage, sauropsids, gave rise to all modern reptiles and birds: lizards, snakes, turtles, crocodilians, dinosaurs, and their descendants. The other lineage, synapsids, is ours. Synapsids developed a single opening, low on each side of the skull, positioned to accommodate increasingly powerful jaw muscles. This seemingly minor detail marks a real evolutionary split confirmed by hundreds of other anatomical features. Early synapsids, called pyracolosaurs, dominated Carboniferous and early Permian ecosystems. Some remained small and lizard-like. Others grew to several meters in length. The most famous are sailed-back forms like Dimetrodon, often mistaken for a dinosaur, but actually more closely related to humans than to any reptile. Dimetrodon lived 280 to 270 million years ago and was an apex predator up to 4 meters long. and had differentiated teeth, large canines for piercing and smaller teeth for cutting. This dental specialization foreshadowed the complex tooth patterns that would later characterize mammals. During the Permian periods, synapsids dominated terrestrial ecosystems worldwide. A more advanced group, called therapsids, emerged around 275 million years ago and became the most successful land animals. Therapsids showed clear movement towards mammalian characteristics. Their teeth were highly differentiated: incisors for cutting, canines for stabbing, complex cheek teeth for chewing. Their limbs moved more distinctly beneath their body rather than sprawling to the sides, improving locomotion efficiently. Some developed secondary palates, bony shelves separating mouth from nasal passages, allowing simultaneous breathing and chewing. This reflects increased metabolic demands. An animal that needs constant oxygen intake benefits from being able to breathe while processing food. Then catastrophe struck. At 252 million years ago, the largest mass extinction in Earth's history occurred. Massive volcanic eruptions in Siberia triggered rapid global warming, oceans acidification, oxygen depletion, and ecosystem collapse. 90% of marine species and 70% of terrestrial vertebrates went extinct. The diverse therapsids that had dominated the Permian were nearly wiped out. Our lineage survived as cynodonts, small therapsids, mostly under 50 centimeters long, with advanced features including even more specialized teeth, upright limb posture, and likely elevated metabolic rates. While archosaurs, the group leading to dinosaurs, rapidly diversified and came to dominate Jurassic ecosystems, cynodonts remained small and unobtrusive, but they were accumulating features that would define mammals. The cynodont jaw underwent critical restructuring. In earlier vertebrates, the lower jaw consisted of multiple bones. Cynodonts had one large tooth-bearing bone, the dentary, that expanded backwards, becoming larger and more robust, while other jaw bones shrank. This allowed more precise chewing control. As the dentary took over jaw function, the small bones at the back of the jaw, no longer needed for jaw mechanics, eventually detached entirely and migrated into the middle ear, becoming the malleus and incus. Combined with the staples, evolved earlier from a gill arch, these three bones conducted sound from the eardrum to the inner ear with far greater sensitivity than the single-bone reptilian ear. This transition happened gradually through numerous species over millions of years, with some fossils showing intermediate stages where bones served dual roles in both jaw and ear. Teeth became increasingly complex, with multiple cusps arranged in precise patterns, creating surfaces that could crush, shear, and grind simultaneously. Different cynodont lineages evolved different cusp patterns for different diets, insects, plants, or mixed omnivorous foods. Precise tooth occultation, bringing upper and lower teeth together in exact alignment, required sophisticated jaw control and neural processing. Evidence for fur appears in late cynodonts. Some fossils show tiny pits in snout bones where whiskers attached. Whiskers are specialized hairs, indicating hair existed elsewhere. Some fossils show turbinal bones, delicate scrolled structures in the nasal cavities supporting membranes that warm and humidify inhaled air. Turbinals are necessary in animals with high breathing rates to prevent water loss through exhalation. Their presence suggested high metabolic rates requiring insulation, or fur, to maintain body temperature. Fur only benefits animals producing substantial internal heat. The evolution of fur implies a transition towards endothermy, internal temperature regulation. Endothermy is metabolically expensive, requiring much more food, but provides enormous advantages. Activity, regardless of external temperature, sustained high-speed movement, and the ability to inhibit cooler climates or be active at night when cold-blooded animals are sluggish. Lactation likely evolved during this period as well, though mammary glands don't fossilize. The logic is straightforward. Endothermic metabolism is energetically expensive, and newborns face severe challenges. They're tiny, lose heat rapidly, and can't find food independently. Providing nutrient-rich secretions dramatically improves newborn survival. The precursors probably existed as glandular secretions moistening eggs, similar to fluids keeping amphibian eggs moist. Over time, these secretions became more nutritious and essential, eventually becoming true milk. By 210 million years ago, in the late Jurassic, one small-bodied cynodont lineage showed the most advanced features: precise dental occultation, complete secondary palates, large brain case indicating expanded brains, small body size suggesting high metabolic rates, and skeletal features indicating agile, possibly nocturnal lifestyles. These mammalia lifeforms lived in forest undergrowth, possibly eating insects and small prey, active primarily at night when they faced less competition from dinosaurs. Nocturnality drove evolution of enhanced hearing and smell, both characteristic mammalian traits. By 200 million years ago, in the latest Jurassic and earliest Jurassic, true mammals existed. They possessed fur, lactation, endothermy, 3 middle ear bones, precise dental occulation with complete tooth patterns, single-boned lower jaw articulating directly with the skull, large brains, and 4-chambered hearts. These early mammals remained small. Most were shrew or mouse-sized, and nocturnal, avoiding the dinosaurs dominating Mesozoic ecosystems. Mammals diversified into 3 major groups: monotremes, which retained egg-laying, marsupials, which evolved live birth with immature young completing development in pouches, and placentals, which evolved sophisticated placentas allowing longer internal development and more mature offspring. Our lineage belongs to the placentals. Through 150 million years of the Mesozoic, the age of dinosaurs, our ancestors remained small, mostly nocturnal mammals, evolving acute senses, sophisticated brains, and complex behaviors, waiting in the ecological margins for an opportunity that would come only when an asteroid removed the dinosaurs 66 million years ago. For 150 million years, our ancestors lived as small, mostly nocturnal mammals in a world dominated by dinosaurs. The largest Mesozoic mammals rarely exceeded the size of a modern badger, and most were far smaller, shrew-sized insectivores, scurrying through underbrush, climbing trees, or burrowing underground. They had fur, produced milk, maintained constant body temperature, and possessed acute hearing and smell suited for nighttime activity. But they remained ecological opportunists, confined to niches that giant reptiles didn't occupy. Everything changed 66 million years ago when a 10-kilometer asteroid struck what is now Mexico's Yucatan Peninsula, triggering the Cretaceous-Paleogene extinction that eliminated non-avian dinosaurs and approximately 75% of all species. The removal of dominant herbivores and predators created a sudden, dramatic opening in ecosystems worldwide. Mammals, which had spent the Mesozoic evolving sophisticated sensory systems, complex brains, and behavioral flexibility, were positioned to exploit this opportunity. The immediate aftermath was catastrophic: impact winter, widespread fires, and ecosystem collapse. But mammals had advantages that aided survival through the crisis. Small body size meant lower food requirements. Endothermy allowed activity even when temperatures dropped. Many were omnivorous or insectivorous, feeding on food sources that persisted through the extinction. Burrowing species sheltered underground. Within a few 100,000 years after the impact, mammal diversity began expanding rapidly into ecological roles previously held by dinosaurs. Early Paleothean mammals, in the first few million years after extinction, remained relatively small but began diversifying in diet and habitat. Some lineages became specialized herbivores, developing teeth adapted for grinding tough plant material. Others became carnivores, evolving shearing teeth and predatory behaviors. Some returned to water, beginning the lineage leading to whales. Others took to the air, becoming bats. Our specific lineage moved into trees. The primates emerged around 60 million years ago during the Paleocene, originating from small insectivorous mammals that had already been climbing. The earliest primates, called Plesioadaphomians, lived in the forests covering much of North America and Europe during the warm period. They looked somewhat like modern squirrels, small, agile, with claws for gripping bark. but they were begging to develop features that would define primates. Forward-facing eyes providing binocular vision, grasping hands with mobile fingers, and brains with enhanced visual processing. The transition to tree-dwelling life created specific selective pressures. Moving through a three-dimensional arboreal environment, where a misstep means a potentially fatal fall, strongly favors accurate depth perception and precise motor control. Forward-facing eyes with overlapping visual fields allow stereoscopic vision. The brain processes slightly different images from each eye to calculate precise distances. This is far more useful for judging the distance to the next branch than the wide field vision of animals living on flat ground who need to watch for predators approaching from any direction. Grasping hands evolved from holding onto branches. Early mammals had claws for digging or climbing on bark. Primates developed nails instead of claws, with sensitive pads on the fingertips underneath. The fingers became more mobile, with multiple joints allowing complex movements. Eventually, some primates evolved opposable thumbs that could press against the other fingers, creating a precision grip, allowing manipulation of small objects. This seemed like a simple adaptation for holding branches and picking fruit, but would later enable tool use in our lineage. The brain expanded to process complex sensory information and coordinate movement through three-dimensional space. Tree dwelling requires constant spatial awareness, tracking your position relative to branches, judging distance accurately, planning movement sequences where each hand and foot must reach the right location in the right order. The cerebellum, coordinating movement, expanded. Visual cortex, processing spatial information, also enlarged. Prefrontal regions, planning and decision-making, also developed further. By the Eocene, around 50 million years ago, true primates with all these characteristic features, forward-facing eyes, grasping hands with nails, enhanced brain size, were widespread and diverse. They split into two major groups: strepsitohenes, which include modern lemurs and lorises, and haplorhenes, which include tarses, monkeys, apes, and humans. Our lineage follows haplorhenes. Haplorhenes show several advantages over strepsitohenes. They rely more heavily on vision and less on smell. The snout shortens, and olfactory regions of the brain reduce, while visual regions expand further. They have a bony wall completely separating the eye socket from the jaw muscles behind it, protecting the eye and allowing the eyeball to move more freely. Their pregnancies last longer and produce fewer but more developed offspring, with expended periods of infant dependency during which parents invest substantial care in teaching. Around 40 million years ago, during the Eocene, our lineage existed as small haplorhene primates living in tropical forests, probably resembling modern tarsiers, large-eyed, agile tree dwellers eating insects, small vertebrates, and fruit. They were social, living in small groups, where individuals recognized each other, maintained relationships, and cooperated in raising offspring and watching for predators. Social living created new selective pressures. Individuals who could read social cues, predict other behaviors, and form strategic alliances and navigate complex group dynamics left more descendants than socially inept individuals. This drove further brain expansion, particularly in regions processing social information. The anthropoids, monkeys, and apes, emerged from these early haplorhenes around 40 to 45 million years ago, possibly in Africa or Asia. Anthropoids are distinguished by larger body size, larger brains relative to body size, and increased reliance on vision over smell, longer juvenile periods with extended parental care, and more complex social structures. They split into Old World monkeys in Africa and Asia, and New World monkeys in South America. The groups separated when continental drift widened the Atlantic Ocean. Our lineage belonged to Old World anthropoids. By 30 million years ago, in the Oligocene, our ancestors were monkey-like primates living in African forests. They were fully diurnal, active during the day, with excellent color vision. Color vision is especially useful for primates eating fruit because it allows distinguishing ripe, nutritious fruit from unripe or spoiled fruit at a distance. The shift to diurnal activity made possible because mammals no longer faced competition from large diurnal dinosaurs, allowed primates to exploit food resources available during daylight. Around 25 million years ago, the line leading to apes split from Old World monkeys. Early apes were larger than most monkeys, with more robust skeletons, no tail, and modified shoulders allowing greater arm mobility. The shoulder changes reflects a shift in locomotion. While monkeys typically run along branches on all fours, apes began using their arms for suspensory locomotion, hanging beneath branches and swinging between them. This requires mobile shoulder joints rotating through a wide range of motion. Suspensory locomotion is energetically efficient in forests where trees are spaced apart and fruit is distributed in terminal branches too thin for large animals to walk on. An ape can hang beneath a branch too weak to support walking weight, reach far out to pick fruit, and swing to the next tree without descending to the ground and climbing again. This locomotion style shaped the ape body plan. Broad chests with shoulder blades positioned on the back rather than sides, highly mobile shoulder joints, strong arm relative to legs, and shortened, stiffened spines providing stability while hanging. The Miocene, from 35 to 5 million years ago, was the golden age of apes. Dozens of ape species lived across Africa, Europe, and Asia, ranging in size from small, gibbon-like forms to much larger animals. Climate during the early and middle Miocene was warm and wet, with extensive forests. But around 8 to 10 million years ago, global cooling and drying reduced forest coverage, particularly in Africa and Eurasia. Many ape lineages went extinct. Others retreated to remaining forests. Our lineage during the late Miocene existed as large-bodied apes living in African forests. These were the last common ancestors of modern great apes and humans. They probably resembled modern chimpanzees in some ways. Strongly built, intelligent, socially complex, capable of both tree climbing and ground movement. They ate primarily fruit, supplemented with leaves, insects, and occasionally meat from hunting small animals. By 7 to 8 million years ago, our lineage had split from the lineage leading to chimpanzees, our closest living relatives. The split was driven by geography. Climate change and geological activity fragmented forests, separating populations. One population remained in dense forests and became the chimpanzee line. The other population occupied more open wetland and forest edge environments, where trees existed but in patches rather than continuous canopy. This environmental difference created the selective pressures leading toward human-specific adaptations. The forest edge population faced different challenges than forest-dwelling apes. Food was more patchily distributed, requiring traveling longer distances between feeding sites. Open spaces between tree patches meant increased predator exposure. Standing upright to see over tall grass and traveling efficiently across open ground became advantageous. This population began spending more time on the ground and less in trees, settling the stage for the most distinctive human adaptation, habitual bipedalism, walking upright on two legs as the primary mode of locomotion. By 6 to 7 million years ago, our lineage consisted of apes showing the earliest signs of bipedal adaptation. Fossils from this period are fragmentary, but they show changes to the skull base, leg bones, and pelvis, suggesting these animals could walk upright, though probably not as efficient as later hominins. They retained long arms and curved fingers suited for climbing, indicating they still spent significant time in trees. They lived in African woodlands, eating fruit, leaves, and probably using simple tools like sticks to extract insects or crack nuts. Behaviors seen in modern chimpanzees and likely present in the last common ancestor. The stage was now set for the hominin radiation, the diversification of bipedal apes that would eventually produce modern humans. These animals stood at a threshold. They had large brains for their body size, complex social structures, flexible diets, and a body plan beginning to adapt for upright walking. The specific sequence of changes, full bipedalism, dramatic brain expansion, tool manufacture, language, and culture, happened over the next 6 million years, through a series of species we'll trace in the next chapter. But the foundations were established here in the Miocene. apes responding to environmental change by spending more time on the ground and beginning the transformation from forest-dwelling frugivore to savanna-crossing, tool-using, socially complex hominin. Our lineage now exists as bipedal apes, living in African woodlands and forest-edge environments around 6 to 7 million years ago. These animals walk upright habitually, not occasionally like chimpanzees, but as their primary mode of locomotion, yet retain long arms and curved fingers, indicating they still climb trees regularly. They have brains slightly larger than chimpanzees relative to body size, complex social groups, and the cognitive capacity to use simple tools. Over the next 6 million years, this lineage will undergo dramatic changes. Full skeletal adaptation to bipedalism, brain size tripling, dependence on stone tool technology, evolution of language, and development of cumulative culture. This chapter traces that transformation through a series of species, each showing incremental changes that collectively created modern humans. The earliest confirmed hominin, a member of the human lineage after the split from chimpanzees, is Sahelanthropus tudensis, dated to 7 to 6 million years ago from Chad in Central Africa. The fossil is a skull showing a mix of ape-like and human-like features. The face is relatively flat, unlike the protruding face of chimpanzees. The canine teeth are smaller than chimpanzee canines. Most importantly, the foramen magnum, the hole where the spinal cord enters the skull, is positioned toward the bottom of the skull rather than toward the back, indicating the head balanced atop a vertical spine. This is the signature of upright walking. Why bipedalism evolved remains debated, but the advantages in woodland savanna environments are clear. Walking upright is more energy efficient than knuckle walking when covering long distances across open ground. It frees the hands to carry food, tools, or infants while traveling. It raises the head higher, improving sight lines over tall grass for spotting predators or locating food resources. It reduces sun exposure. A smaller surface area faces directly overhead sun, helping with heat regulation. These advantages weren't individually transformative, but collectively they made bipedalism beneficial enough that natural selection favored individuals with skeletal changes, improving upright walking efficiency. By 4.4 million years ago, our lineage consisted of Ardipithecus ramidus, known for extensive fossils in Ethiopia. Ardipithecus was fully bipedal when on the ground, with a pelvis showing clear adaptations for upright walking. The pelvis was shorter and broader than in apes, repositioning hip muscles to stabilize the body during the single-leg stance phase of walking. The foot had a divergent big toe, more mobile than in later hominins, still useful for grasping branches, but the other toes and the ankle showed adaptations for bipedal walking. Ardipithecus was small, about 120 centimeters tall and 50 kilograms, lived in woodland environments, and ate primarily fruit, leaves, and nuts. The Australopithecus genes appeared around 4 million years ago and diversified into multiple species between 4 and 2 million years ago. The most famous is Australopithecus afarensis, known from hundreds of fossils, including Lucy, a 40% complete skeleton dated to 3.2 million years ago. Australopithecus afarensis was clearly bipedal, with no remaining doubts. The pelvis was fully adapted for upright walking. The legs angled inwards at the knee to position feet under the body's center of mass. The feet had lost the divergent big toe and developed a longitudinal arch for efficient push-off during walking, and the spine had developed the S-curve characteristic of humans that positioned the head over the body's center of gravity. Australopithecus still had relatively small brains, about 450 cubic centimeters, roughly chimpanzee-sized, and relatively large teeth with thick enamels suited for chewing tough plant foods. They lived in environments ranging from woodland to more open savanna, traveled long distances on foot, and probably still climbed trees for safety at night or to access certain foods. Stone tools had not yet been found clearly associated with Australopithecus, though they may have used unmodified stones as hammers or anvils, and certainly used perishable tools like sticks that don't preserve. Around 2.8 million years ago, the first members of our own genus, Homo, appeared in East Africa. The earliest species, Homo habilis, showed key advantages over Australopithecus. Brain size increased from 600 to 700 cubic centimeters. Teeth and jaws became slightly smaller, and crucially, Homo habilis is associated with the first definitive stone tools, the Oldowan industry, consisting of simple flaked stones creating sharp edges for cutting meat and processing plant foods. Stone tool manufacture requires understanding that striking one stone against another at a correct angle will detach a flake with a sharp edge. This seems simple, but requires cognitive abilities most animals lack. The ability to envision a desired outcome, plan a sequence of actions to achieve it, and possess fine motor control to execute precise strikes. Tool use isn't unique to humans. Chimpanzees use sticks to fish for termites. Sea otters use rocks to crack shells. But manufacturing tools to a standardized pattern that's taught and transmitted across generations represents a cognitive threshold our lineage crossed around 2.6 to 2.8 million years ago. Tools changed everything. Sharp stone flakes allowed access to carcasses, cutting through thick hide to reach meat that would be inaccessible otherwise. Access to meat, a calorie-dense and nutrient-rich food source, provided the energetic resources to support larger brains. Brains are metabolically expensive tissue, consuming about 20% of resting metabolism in humans, despite representing only 2% of body weight. The shift towards higher quality diet, including meat and bone marrow, made this expense sustainable. Homo erectus appeared around 1.9 million years ago and represents another major transition. Brain size increased to 900 to 1100 cubic centimeters. Body proportions became fully modern. Tall, long legs, short arms, narrowed pelvis, and barrel-shaped chests, all adaptations for efficient long-distance walking and running in open environments. Homo erectus manufactured more sophisticated tools, the Acheulean industry, featuring symmetrical hand axes carefully shaped on both sides, requiring planning and mental templates. They controlled fire by at least 1 million years ago, using it for cooking, warmth, light, and protection from predators. Fire and cooking are transformative because cooking pre-digests foods, breaking down tough plant fibers and denaturing proteins, making nutrients more accessible and reducing the energy needed for digestion. Cooked food can be chewed and digested faster than raw food, allowing further reduction in jaw and gut size while still extracting sufficient nutrients. The energy saved on digestion could be allocated to brain growth and maintenance. Homo erectus was the first hominin to leave Africa, spreading into Asia and reaching as far as Georgia, China, and Indonesia by 1.8 million years ago. This required cognitive sophistication for navigating unfamiliar environments, solving novel ecological challenges, and possibly coordinating group activities through some form of communication beyond what earlier hominins possessed. Homo erectus persisted for over 1.5 million years, making it the longest surviving hominin species beside our own. Between 800,000 and 300,000 years ago, Homo erectus populations in Africa evolved into forms transitional between Homo erectus and later hominins, sometimes called Homo heidelbergensis. Brain size increased to 1,200 to 1,300 cubic centimeters, approaching modern human size. These populations manufactured even more sophisticated tools, constructed shelters, and may have begun using symbolic markers, orche pigments whose purpose isn't clear but may relate to decoration or communication. Around 300,000 to 400,000 years ago, hominin populations in Africa, Europe, and Asia began diverging into distinct lineages. European and Western Asian populations became Neanderthals, with brains as large as or larger than modern humans. Sophisticated stone and bone tools, evidence of caring for injured group members, and burial of the dead suggest symbolic thought. Eastern Asian populations became Denisovians, known primarily from DNA and few fossil fragments. African populations became anatomically modern Homo sapiens. The oldest fossils of Homo sapiens with fully modern anatomy date to around 300,000 years ago from Morocco and Ethiopia. These early modern humans had high, rounded skulls, housing brains averaging 1,350 cubic centimeters, small faces with reduced brow ridges, prominent chins, and fully modern body proportions. But early Homo sapien behavior wasn't immediately modern. For over 200,000 years, they manufactured stone tools not dramatically different from those made by contemporary Neanderthals or late Homo erectus populations. The transition to behavioral modernity, the full suite of behaviors characterizing recent and living humans, happened gradually between 200,000 and 50,000 years ago. Archaeological evidence shows appearance of symbolic artifacts, shell beads used as ornaments, orchid pigmentations for decoration, engraved patterns on stones and bone suggesting abstract thought, more sophisticated and diverse tool types including specialized tools for specific tasks, long-distance trade networks moving rare materials hundreds of kilometers, and eventually, around 40,000 years ago, representational art including cave paintings and carved figurines. Language almost certainly evolved during this period, though its origins remain unclear because a language doesn't fossilize. The anatomical prerequisites existed. Homo sapiens had a descended larynx, allowing production of diverse vowel sounds. Neural control of breathing allowed sustained vocalization, and brain regions specialized for language production and comprehension. Genetic evidence shows modern humans possessed versions of genes like FOXP2 involved in language ability that differ from versions in Neanderthals, suggesting language-related evolution in our lineage within the last few hundred thousands of years. Language is transformative because it allows the transmission of complex information between individuals and across generations without requiring each individual to learn everything through personal experience. A group with language can accumulate knowledge, how to make specific tools, where seasonal food sources are located, which plants are medicinal, how to navigate terrain, and pass the knowledge on to children verbally. This creates cumulative culture, where each generation builds on previous generations' innovations rather than starting from scratch. Around 70,000 to 60,000 years ago, a relatively small population of Homo sapiens, perhaps a few thousand individuals, left Africa and began spreading into the Middle East, Asia, Europe, and eventually Australia and the Americas. This expansion happened in waves over tens of thousands of years. When Homo sapiens encountered Neanderthals in Europe and Western Asia, the groups interbred. Modern non-African humans carry 1 to 2% Neanderthal DNA. Similarly, inbreeding with Denisovians occurred in Asia, with some modern populations carrying Denisovian DNA. By 40,000 years ago, Homo sapiens was the only surviving hominin species. Neanderthals went extinct around 40,000 years ago, possibly outcompeted by Homo sapien populations with slightly more efficient hunting strategies, more sophisticated social networks, or technological innovations. Denisovians disappeared around the same time. Late surviving populations of small-bodied hominins, like Homo floresiensis in Indonesia, persisted until perhaps 50,000 years ago before vanishing. The last 40,000 years saw Homo sapiens spread to every continent except Antarctica, adapting to environments from Arctic tundra to tropical rainforests to high-altitude plateaus. Population size grew from perhaps a few million globally 10,000 years ago to over 8 billion today. Technology accumulated exponentially. Agriculture beginning 10,000 years ago, metalworking, writing, cities, industrialization, and now digital technology. Social organization grew from small bands of related individuals to chiefdoms, states, and global interconnected societies. But genetically and anatomically, we remain essentially unchanged from the Homo sapiens that left Africa 60,000 years ago. Our brains are the same size, our bodies have the same structure, our genes differ by fractions of a percent from those ancient populations. What changed isn't our biology, but our culture. The accumulated knowledge, technology, social structures, and symbolic systems transmitted through language and teachings across generations. This is the endpoint of our lineage. From self-replicating chemicals in hydrothermal vents 4 billion years ago, through the first cells, through eukaryogenesis, through early animals, through fish and developing jaws, through the transition to land, through synapsids becoming mammals, through primates entering trees, through apes returning to the ground, through bipedal hominins expanding across Africa, to modern humans spreading globally and building civilizations. Every feature you possess, your cells with mitochondria and nuclei, your backbone and 4 limbs, your warm blood and body hair, your forward-facing eyes and grasping hands, your large brain and capacity for language, traces back through this unbroken line of descent, each feature arising in response to specific challenges faced by specific ancestors in specific environments. You are the current result of 4 billion years of continuous evolution, the temporary endpoint of a lineage that has never once been broken since the origin of life itself. So, 4 billion years. Here we are. You just watched your entire family tree from the bottom to the top with nothing left out. Every ancestor who had to survive, every generation that had to succeed, every near-miss extinction that almost ended your lineage before it could continue. And if you're sitting there thinking, that was long, or that was complicated, you're right, it was. Because the story is long and it is complicated, but that's exactly why it matters that you saw the whole thing. See, when we only learn evolution in pieces, it's easy to wave your hands and say, and then fish became land animals, or eventually we got to humans. It sounds simple. It sounds obvious. Maybe it even sounds inevitable. But you just watched it actually happen. You saw how every single step required something specific to already be in place. How lungs couldn't exist until swim bladders existed first. How jaws couldn't exist until gill arches existed first. How tools couldn't happen until hands existed first, which couldn't happen until limbs existed first, which couldn't happen until fins existed first. Nothing appeared out of nowhere. Everything came from something that came before it. That's what makes this story so powerful and so fragile. Because here's the thing about a chain 4 billion years long. It almost broke, many times. The extinction that killed most species 250 million years ago nearly ended your lineage entirely. The asteroid that took out the dinosaurs was pure luck. It could have missed, and if it had, you probably wouldn't exist. Different world, different dominant species, and a different story entirely. You're not here because evolution was aiming for you. You're here because an unimaginable number of things happened to go right in sequence for longer than we could really comprehend. That's not destiny or design. It's just what happened. And that makes it more meaningful, not less. When you look at evolution in pieces, it's just facts. Trilobites existed, dinosaurs went extinct, humans showed up. So what? But when you see the whole thing, when you trace that single thread from beginning to end without looking away, you realize you're not separate from this story. You're made of it. Your body is a living record of everything that came before. Your cells still work like bacteria because they used to be bacteria. Your skeleton is still built like a fish skeleton because that's what it started as. Your brain is still running on software that evolved to help small mammals not get eaten. Now trying to do calculus and write poetry and figure out where it came from. That's what this project has really been about. Not just showing you that evolution happened, or even how it happened, but helping you see that you are evolution happening, right now. The chain didn't end, it's still going. You're the current chapter in a story that's been writing itself for 4 billion years. And now, you know the whole story. You've seen where you came from, how you got here, and why every step mattered. You've climbed your family tree, from the roots to the branch you're sitting on right now. This is your history, all of it. From the first molecules that learned to copy themselves, to you watching this video trying to understand how it all happened. Pretty wild when you think about it. Thanks for coming on this journey. Thanks for sticking with us through 3 hours of deep time and weird transitions and extinctions that almost ended everything. You made it, just like your ancestors made it. And now, you know how.